there's no one out there talking about why you need to do a handstand for your longevity, and I think there's absolutely, I'm not saying you need to be able to do handstands to live a long life, but I'm saying there's absolutely something to understand in there that we've barely even tried to start understanding. And this was kind of like, with the LA Veterans Administration trials showing the increase in cancer, and people like Steinberg, who were like chief architects of the cholesterol hypothesis, being afraid of the lipid peroxidation in the LDL particle, they kind of very quietly moved away from promoting corn oil, which is what my grandparents were told to eat, and moved towards olive oil because it was considered neutral, because it doesn't affect cholesterol levels, but it doesn't cause peroxidation, right? One of the most powerful associations with Crohn's disease incidence in a country is its degree of development.
Yeah.
And there is something like a 62-fold difference in the incidence of Crohn's disease between America and the Caribbean.
Tell me who you are and what you do.
Chris Masterjohn. I run a mitochondrial testing company. I come from a background of nutrition science and using the power of nutrition to help people achieve their highest potential in terms of their health.
How do you test mitochondria?
In our case, we send people a kit with a cheek swab, and they swab their own cheek. They send it in, goes to the lab, generates results, goes to our system, generates a report. What we're looking at is the specific enzyme complexes in the mitochondria that are the mitochondrial engines that produce most of your cellular energy in the form of ATP. And if you look at the details of that machinery, you can see patterns, ratios, things that are high and low that tell you about parts of the system that have specific nutritional implications. So you might see a pattern that says, oh, it looks like methyl groups, which are dependent on B vitamins and protein and other things going on, as well as your genes, like the famous MTHFR, are not reaching your mitochondria because that's what's manifesting this specific pattern that makes that the limiting bottleneck for your energy metabolism. And the protocol might then be a methylation-focused protocol, whereas in another case, someone has a pattern that makes it look like they would do better on a high-carb diet or a low-carb diet, or makes it look like they might have a specific need for a specific vitamin like riboflavin or a vitamin-like substance like CoQ10, for example.
Okay, so then this definitely puts, I didn't catch this when I normally preview whoever the guest is going to be, particularly if they've been on a recent podcast. You happen to have been on a recent podcast, so it worked out pretty well for me. You were telling Joe Rogan that mitochondrial function as the root of all health and disease is not getting it finally seems to be getting its due lately, right?
I think it's about to start getting its due.
Okay, but it's, even though a lot of people are chatting about it, even though it's the new hotness, you would say it's not, it's just now starting, like we're just cracking open the book.
Well, to take an example that you're probably familiar with, the predominant treatments to raise LDL receptors are statins and Oreo cookies right now. And no one's thinking about the 5 classes of ATP-dependent motor proteins that allow the LDL receptor to do its function. Because what is it doing is it's bringing cholesterol from outside your cell to inside your cell. What does that involve? It involves movement. So you don't need to know the molecular biology to know right off the bat that if it requires movement, it requires energy. And to reason, oh, the energy must come from the mitochondrial transformation of your food energy into the principal usable form of energy as ATP. But if you go look up the molecular biology of the LDL receptor, you will see that there are a lot of motor proteins that are involved in every stage of its movement to capture the LDL particle and bring it into the cell. So until we have people saying, oh, my cholesterol is high, why, what are the limiting bottlenecks in my energy metabolism to allow the dynamic movement of the cholesterol into the cell, then I don't think people are thinking of it in the front of their minds. They might be thinking of it in the back of their minds, or as footnote or a side note to something, but they're not thinking about it as the central determinant of health.
Yeah, there's no getting around it. We're going to get super geeky here, but I do think for setting the stage, especially for people just dipping their toes in this water, ATP is often described as the currency of the cell, often, right? Because there's quite literally an economy that's going on inside the cell. And the production of ATP is relevant because while it wouldn't be exactly totally categorically energy, because there's kind of an exchange rate, if you will, what you're describing with regard to like things like transport proteins are to do transportation for a protein. Often there's literally a kind of expenditure with ATP usage, right? So for example, when you're talking about the LDL receptor, first of all, just making one requires ATP and a number of chemical reactions. But then on top of that, let's say that we're talking about taking up a lipoprotein. That requires both chaperone proteins and ATP expenditure for those because it's a very active process, right? So for example, taking a SRB1, generating the caveolae that creates this like little bubble, if you will, that you're pulling the lipoprotein into. And then it's not just as simple as now it's there and it'll magically float to the other side of the cell. That actually has to be chaperoned as well. There needs to be these transport proteins that are taking it all the way through. So when we're talking about things like an active process, it does make a lot of sense that we care about, hey, where does this ATP come from? And how important is that to the function of the cell? We could say it's absolutely vital, right? In just about every respect you can think of, that's especially for an active process.
Yeah, I think at the high level, mitochondrial energy production is fueling the production of everything in your body. It's fueling the maintenance of it, the repair of it, and the distribution of it. And so it's intuitive to think about movement implying energy, but just as in our society where we invest enormous amounts of fossil fuel energy in producing all the things around us, we're investing enormous amounts of food energy, and the usable form of that is ATP. And I agree with what you said that there's an exchange rate, and I think an economy is the right way to think about it. So the value of ATP goes up and down depending on the circumstances, but I think but that doesn't change the fact that there's a cost in ATP to running all those processes. A lot of the you have assembly proteins, you have motor proteins involved in putting them in the right place. You've got the cytoskeleton, basically inside the cell, you basically have roadways and train tracks along which everything is moving, and the movement down, if that cholesterol is going to come in and move to a place where it's going to be freed to be used for useful things, that whole transport way is going to be involving ATP hydrolysis the whole way. So ATP is a fairly universal currency of use. But that said, getting back to sort of what you described as part of your business, it sounds like what you're trying to do is also identify deficiencies. Outside of ATP, is the mitochondria getting what it needs, right? Yeah, that's exactly right. So the mitochondria is dependent on all of the nutrients. And so it's always, I do think that people should always be thinking about their mitochondria first when they're thinking about their health. But they should also, from an action standpoint, they should also always be thinking first of the basics of good nutrition, good exercise habits, good, outdoor, indoor exposure, good movement, good sunlight regulation and lighting regulation, all those things. All of those things are important as a baseline to be supporting your mitochondrial function. But there's also an idiosyncratic layer that's unique to each of us. And so your needs are going to be different than mine based on the fact that you have different genetics than I do, based on the fact that you have a different past experience than I do, and even based on the fact that our diets are different right now and our dietary histories are also different. And that will manifest as some things working, better than others in your mitochondria. And so to know what that unique layer, to know if I'm doing everything that I believe I should be doing correctly, to know that there are still parts of my mitochondrial energy production that are limiting me, and to know that tells me something unique about what I need to bring myself to the next level is the next most useful thing for me to know.
But we as humans, we tend to think in these short time spans, right? Like, I want to take supplement X, and then do I feel an immediate result? And so by the same token, what I want to do to improve the health of my mitochondria, I might be like, okay, so what will I see in like 2 weeks, right? But it's really a long game. I mean, that's certainly something that I think is an ongoing theme in the materials that I've watched you, particularly when you were talking about it on Joe Rogan. A lot of times you're optimizing for a much longer time span, which makes a lot of sense.
I think as a default, you want to be optimizing on long time horizons, but you do sometimes see short-term miracles. So as an example, one of the early customers we had with the Mitome test, it identified a specific need for CoQ10 in her. And now at the basics, there's a lot of randomized controlled trials on CoQ10 supplementation, and they generally paint a picture where 1 to 200 milligrams per day benefits the average person moderately with respect to a little bit better glucose metabolism, a little bit better blood pressure, etc. But they also suggest that there aren't enormous miracle effects, and they also suggest that a little bit higher than that and you're going to be in trouble. So for example, if you look at the dose responses within those trials, glucose and blood pressure get better at 1 to 200 milligrams of CoQ10 a day, and then they start to get worse after that. You lose the benefit, and then you get into a point where your blood pressure or your glucose is higher than baseline. Now, anyway, this customer, Jacqueline, she had not had her period for over 10 years. She was in her early 20s when that started, so it's not menopause.
Can I ask, was she underweight?
She was not underweight. I don't know if she was underweight when it started, but she wasn't underweight at the time that we were talking, or at the time she had the test. Anyway, her test revealed a blockage that looked like she should be trying 700-800 milligrams of CoQ10. And so she did that, and she asked me, do you think I'll get my period back? And I said what you just told me, which is, well, look, you're playing the long game. Like the fact that it's been gone for 10 years suggests that it's not going to come back in 2 weeks. And this is, maybe a year or two. But in general, my baseline position is, if you've let a problem develop for X amount of time, you should expect to take X amount of time to dig yourself out of it, right? But anyway, 2 weeks later, she sends me an email and she says, I got my period. And that for the first time in 10 years. And that was a year ago. And everything, is still the way it was at the 2 week mark for her. So you see those miracles like that. And I think that speaks to that idiosyncratic layer, right? Like I think health is kind of like a 2-layered cake. Like the bottom half of it, cake's not maybe
the best analogy.
But yeah, whatever. Anyway, 2-layered quiche, right? So anyway, the bottom of it is the things that we should all be doing for our health, and these are, there's good descriptions and bad descriptions about what that is, but in general, there is some chunk that is common to what humans should be doing to have good health.
You should get some exercise. You should get some sunlight. You should. Those are like everybody needs to have some amount, right?
Yeah, I mean, I think you could certainly go full bore into the details of what that means, but the basics I would say are good nutrition, good exercise habits, good sleep routine, good stress management, good sunlight exposure. Those are kind of the big basics that stand out, and much could be said about any of those.
I do kind of want to just say, of the buckets that you just mentioned, one of them is so complex, the nutrition one. I feel like the degree of depth of both knowledge, including what we just already know versus what we still need to learn, with, say, exercise, we still have lots that we could learn, but not compared to nutrition. I feel like nutrition, we're still learning quite a bit, and we're also discovering what we're doing wrong more recently, right? But yeah, like exercise, sleep, I feel like we're learning a lot about sleep, but as far as just total amount of sleep, trying to get enough REM sleep, enough deep sleep, and so forth, still much to learn. But as far as hitting what's required and avoiding deficiency, I feel like that's more knowable relative to the degree of complexity and uncertainty that we have right now with nutrition. Would you agree with that?
I don't know. I would say that, I do think that in terms of elite athleticism, we have really good bodies of knowledge in a bunch of sports, and we could invent new sports that would require, expansion of that knowledge, but it's going to all be determined by existing knowledge of biomechanics. So in terms of what we could know to support elite athleticism across the domains of sports, there's probably not a lot to learn there. But I think that what should be taken from that for the average person to be healthy, I think that needs a lot of work.
Right. And to emphasize, I'm just talking about the average person. I'm saying if you had a class.
Well, no, I'm saying that's where we have the worst knowledge. So, and I think the worst practice as well. Like I think that we're at the level of saying that, everyone should do X amount of cardio, and we're kind of shifting into the world of, and should also add 2 or 3 days of resistance training. And I think that's barely scratching the surface of what the average person should be doing. I mean, I take this from, look at the longevity space, right? So there's an enormous obsession with cardiorespiratory fitness, and it's well-deserved, in that cardiorespiratory fitness is a determinant of longevity. But there was a study that came out last year that looked at, out of all the elite male athletes, who lives the longest. And the cyclists and runners who should have really good cardiorespiratory fitness, they have 2 or 3 years on the average person in the population. But the pole vaulters and gymnasts have 8 years. And so what that tells me is that, and the weightlifters are down further than that, right? So what that tells me is that we've cracked the code on cardiorespiratory fitness being important, but it isn't actually that important in terms of the broad spectrum of what you could do for exercise. And so now I can't tell you with any kind of certainty that if you become a gymnast, you're going to live 8 years longer than the average
Right.
person.
We'll get into causality.
But
yes.
But I think that should stop and make us ask the question: are there other components of exercise that play a role in longevity that are more important than cardiorespiratory fitness? And I think that there are, and I think that gap is where there's no one out there talking about why you need to do a handstand for your longevity, and I think there's absolutely, I'm not saying you need to be able to do handstands to live a long life, but I'm saying there's absolutely something to understand in there that we've barely even tried to start understanding.
With one important caveat that I'm sure you'd agree with, which is that the diets of all athletes are also very different from one to the next. So, for example, with cyclists, runners, particularly in like the 90s and 2000s, maybe it's changed a little bit more recently. Things like, say, for your endurance run, you're going to be having goos while you're, trying to move it across so that you could have enough energy for something that's long distance. Things along those lines would just be a very different part of the regimen of somebody who's doing pole vaulting and, gymnastics, relatively speaking. I think there might just be things that are common to the specific diet of a given class of athleticism with regard to what the culture of that, of
there
what those
might be,
elites
but
are.
I would be surprised if that didn't play out in the other domains, right? So like weightlifters also have a different diet from runners,
Right.
but they don't show a longevity benefit because of it.
Right. And that's what makes it fascinating. And to be sure, this is what one of the challenges are when we break out the grabbing one of those buckets like exercise and saying, hey, I want to ask this question. Does a particular kind of exercise increase longevity? And you and I would both agree we're still looking at associational data. We can't say a lot without doing like an actual RCT or something. But that said, applying some Bradford Hill, that is an interesting. The study that you just brought up, pole vaulters and gymnasts, I think you brought this up with Rogan, the height difference is gigantic. Of course, pole vaulters tend to be taller, right? Gymnasts tend to be a lot shorter.
Yeah, the height difference is opposite between them.
Right. But one thing they definitely have in common is a lot of hard landings, right? So,
well, I don't think it's hard landings, man, because martial artists get punched in the face a lot and they don't show a longevity benefit for that. What's interesting is the volleyball players actually have worse longevity, and I think that there's lots to mine from this observation set, and lots of ideas that could come from it. But I think if you look across it, what stands out about the gymnasts and the pole vaulters is they have excellently functioning joints and biomechanics. And I believe that there is definitely a basis for why that would protect against death. I think it would be protective against both autoimmune conditions and cancer. It would be interesting, of course, to ask the question, what are they dying less of? But the greater the longevity benefit, the more you know that it's veering into cancer and neurological disease, because the way that people on a population level, the sequence of death is you die of heart disease first, and if you don't die of heart disease, you're more likely to get cancer.
Right.
And if you don't die of cancer, you're more likely to die of a sort of a more less clear set of neurological diseases that are going to be picked from to put on your death certificate. So basically the death structure is when an economy is kind of recently developed, everyone's dying of heart disease, and as it advances and as affluence allows people who would have died of heart disease to not die, and they advance in age, cancer takes over. And as you push through that, you get into this kind of really diverse array of people instead of everyone dying from the same thing, everyone's dying from different things. The connection to death is a little bit less clear, and they generally have some form of neurocognitive decline associated with it. And so I think neurological training is huge in both gymnastics and pole vaulting. And I think that would be, that if that's your neurological peak, you could decline a lot, and you're not going to be in neurological decline by the general population definitions. Cancer is very interesting. I'm not saying this is hard evidence that being a gymnast protects against cancer, but the reason I find it so interesting is because there's a lot of science in T cell activation, and one of the unknowns in T cell activation was that to activate for a T cell to activate itself, which is necessary to kill cancer or any other anything else that doesn't belong, like a pathogen. For the T cell to activate itself, it's a very energy intensive process, going back to the mitochondrial energy metabolism. But everyone had studied in the field, in this niche field of T cell activation, all the scientists were working on where does the energy come from. And so they're doing all the delta G calculations that you do in, first year general chemistry class, trying to figure out what biochemical reactions are releasing the energy that can fulfill the need to activate that T cell. What it turned out was that it's kinetic energy. And what happens is the T cell has motor proteins inside itself, and it pushes off the extracellular matrix to create that kinetic energy inside itself that then fuels the T cell activation. And one of the things that cancers do to evade T cell activation is they restructure their extracellular matrix to make it harder to push off from. So there's a gap between that and what I'm saying, which is a hypothesis, and I'm in brainstorming mode. But my brainstorm is that the quality of your extracellular matrix that is maintained through excellently functioning biomechanics helps maintain the type of environment that T cells would activate well off of, but not excessively. And so I think that when you have deviations from that, you can get deviations that favor excessive inflammation, or you can get deviations that favor an inability to respond as needed. And I think that could impact the risk of cancer.
In T cells, correct me if I'm wrong, you could argue is the beginning of the adaptive immune response with regard to they're effectively the ones that are starting the encoding process, right? Because they're ultimately bringing it back to then determine with the B cells. Am I remembering this correctly?
In the course of getting sick, you would first have an innate response that is non-specific.
Right.
And that would be from various types of blood cells, like neutrophils, for example.
Macrophages and so forth.
And then that would, over the course of a few days, switch over to B cells and T cells as part of the adaptive response. And the T cells play various roles that include activating B cells, activating other T cells, or killing things directly.
But getting back to your theory with regard to cancer, because the extracellular matrix, the health of the extracellular matrix, you would argue is part of the relevance to the inability of the T cells activation to get that cascade going. Am I understanding that correctly?
Well, we don't have a lot of data to kind of quantify the magnitude or even the direction on this, but we know that it is possible to have an extracellular matrix that sabotages T cell activation, and we know that because that's part of a cancer strategy, is to modify the extracellular matrix in that way. And we also know that you can have pro-inflammatory effects of the exact same nature. So over the last couple of years, one mystery in Crohn's disease has been, why is the one dietary strategy that is at least as and possibly more effective than top of the line biologics that are, niche drugs that kill parts of the immune system to lower the inflammation? Why is that diet the liquid diet made from Ensure? And, when you look at the quality of the diet, you're like, well, it sure as hell ain't the quality of the diet. So there was an animal experiment last year, or I think this was in 2024 actually, where they showed evidence that the mechanism of the liquid diet is that it reduces the amount of pressure inside the intestines, and that the pressure pushing out is what creates the inflammation in the subsequent segment of the intestines. And it's my suspicion, there's no data on this, but my suspicion is that the part of the reason that a Crohn's patient cannot tolerate the pressure of the food, of normal food, part of it relates to genetic predispositions to inflammatory states and metabolic dysfunction. But I think what you would find if you examined many Crohn's patients is that they all have idiosyncratic biomechanical problems that interact with that. So I think the biomechanical problem is not what causes the person to have Crohn's or not, but I think the biomechanical problem is often what causes the Crohn's to be in one segment of the intestines versus another. And the outer pressure from the food is running up into poorly structured things on the outside that create the inflammation. Another interesting tidbit kind of supporting the pressure hypothesis, well, two others, are that one of the main genetic drivers of Crohn's is turning out to be the gene that encodes a pressure receptor. So it's how many tissues respond to physical pressure. PISO1, that's one of the genes that's associated with Crohn's disease, but also one of the main determinants of when does a person who has Crohn's get a flare is, have they recently traveled to a place that's over 5,000 feet altitude? And that's what that is the reduction of the external pressure, right? So you have too much internal pressure and not enough external pressure is driving Crohn's disease. And the person who found that epidemiological association then did an experiment, might have been this year that it was published, where he took a bunch of Crohn's patients and he put them in a hypobaric chamber for 3 hours.
Oh.
And 33% of them got a flare just from being in the hypobaric chamber for 3 hours.
Okay. Huh.
So this internal external pressure battle is like the unspoken cause of Crohn's disease. Not that it's the cause, it's just, there's 3 or 4 big causes and that's the one that gets the least attention. But what that shows you is that this pressure relationship to inflammation goes both ways. So you have the cancer modifying the external environment to sabotage the ability of the T cell to activate using biomechanical pressure. And you have Crohn's disease being a total dysfunction of the ability to regulate biomechanical pressure in a specific segment of the intestines. One of those is pathologically anti-inflammatory, the cancer. You cannot mount the inflammatory response to kill it. And the other is pathologically pro-inflammatory, Crohn's, where you just cannot get rid of this inflammation in the segment of the intestines. So I look at those and that tells me that there's just this gold mine to be rushed and mined to get information that we have no clue about on this question of how do biomechanics, in a way that goes way beyond cycling and running, cardiorespiratory fitness, how do those biomechanics help us and hurt us on the path to being free of inflammation, being able to mount the right response with inflammation when we need it?
Yeah. So a number of things to unpack there. One, this is an ongoing theme within this podcast, of course, and I think just in the field of nutrition in general, which is how much does the modern food environment set us up for things like Crohn's disease, right? So a lot of diseases are thought in a default way to be genetic. Like, oh, it's bad luck of the draw. You got X. And then a particular food, a particular diet turns out to be relieving for the symptoms of that thing, right? Crohn's disease being one of the common examples that people on a keto diet like to point out. Hey, did you know about keto for Crohn's disease? And then the question then comes back around to, well, now wait a sec, were there things about what the person who had Crohn's disease were on before the diet that they had before that might have set them up to then get Crohn's disease. There's that, and could it be combinatorial? Could it be, what is it? Genetics loads the gun and environment pulls the trigger, right? And then can it be something like, say, epilepsy with regard to like the Charlie Foundation based on Charlie, the son from the famous producer, where he has epilepsy up until, I want to say, his 20s. Being on a ketogenic diet is relieving it, and then he seems to just fully resolve it. And then that's it. And then he can seemingly have any kind of diet past that point. I bring up all of these examples because getting back to Crohn's in particular, I want to pause and think, okay, how much did Crohn's come up before the modern food environment where you and I are used to now? Like, how often was it coming up, say, 100 years ago? Do you know that?
It was probably non-existent. I mean, one of the most powerful associations with Crohn's disease incidence in a country is its degree of development.
Yeah.
And there is something like a 62-fold difference in the incidence of Crohn's disease between America and the Caribbean. So there's like the ecological evidence is not a good way of making inferences about what causes something in a person, but it says a lot about it can say a lot about the society and the societal norms and the things that are constants in that society, So I do get the impression that you would not have any Crohn's if you didn't have ultra processed foods at all. And I'm sure there are nutritional influences on development, but a major risk factor is breastfeeding. So breastfeeding for less than 6 months, which is completely inconsistent with the historical practice of our species, is associated, breastfeeding for more than 6 months has a 80% lower incidence of Crohn's disease than breastfeeding for less than 6 months.
Wow.
Yeah.
That's a strong association. Goodness.
Yeah.
So this will actually be a good place to segue into, usually with guests, I go into their biography first. We're kind of going backwards for a sec. But the reason this was a good segue was how, when I was first coming into this space, I was coming in 2015. And I would say that was when there was kind of an overlap between something that was big dietarily before, that had more of the zeitgeist, more of the attention, and that would be the paleo movement, and a kind of overlap with low carb. And there was even a lot of people on the paleo side who were kind of moving into the low carb side. Paleo, probably one of the biggest conferences at the time, was Ancestral Health Symposium, I want to say. Then there was another one that was big, Paleo Effects, if I'm not mistaken. Ancestral Health Symposium, though, I think is still going, isn't it? Hasn't it been? I know it's kind of petered a little bit, but it's...
Yeah, I'm not sure when they had the last one,
But you kind of came up in that space, right? You were a regular speaker at many of the conferences, and a lot of what you were writing at the time was related to... a lot of the content you were generating was connected to paleo, or at least you were adjacent, if you will. But fun fact, some of my early learnings of lipids came from Chris Masterjohn. You actually wrote a lot on LDL and cholesterol, particularly given that there were a lot of people in paleo who were concerned that if they were eating a meat-heavy diet, that they would get a lot of high cholesterol. So I've got a fun question for you, okay, which is, how much would you say your opinion has changed from those days of learning about paleo? And for that matter, how much has your diet changed versus what your opinion was before?
Well, I think paleo adjacent is a good way to describe me, because I was never deep into the paleo zeitgeist myself. And I actually didn't know that paleo was a thing outside of Lauren Cordain's book until I started looking at my blogger statistics, and I saw that all my traffic was coming from these paleo blogs. And so I kind of discovered that world that way. I did follow Stefan Guillenet at the time, and he also was kind of paleo adjacent. He was more paleo. He was more in the thick of paleo than I was, just in terms of his perspective. But so yeah, so I think a lot of people liked my writings because of this controversy over animal fat. And that's why my writings became popular in the paleosphere. And then I kind of, I found that sphere, and I did speak a lot at the Ancestral Health Symposium. I came from more of a Weston Price background than paleo, and I think there's a lot of agreement and harmony between those two perspectives. But Weston Price was much more focused on what went wrong in the Industrial Revolution, and paleo is much more focused on what went wrong in the Agricultural Revolution. And so Weston Price people were always looking back at what did your great grandparents eat? And paleo people were looking back at what were we eating 10,000 years ago?
Right.
And what they share in common is that they don't eat the way people eat today.
Right.
So there's a lot of agreement that can take place there, but I've always believed that the big harm came in the Industrial Revolution more than the Agricultural Revolution. I do think I've always thought that the paleo movement took their analysis of the archaeology too far in a couple of ways. One is that you can't know the health of a population from the archaeology, and the reason is that you have intrinsic selection bias in the archaeological remains. But more to the point, I think if you look at what happened at the agricultural transition, it's things that aren't even applicable anymore. So everyone got shorter, but everyone in our society is taller than extant hunter-gatherers. So how do you square that circle? But even in the archaeological record, what it looks like is things got really, really bad for a few generations, and then they bounced back up and they regressed to the mean. And my interpretation of that was, you start growing wheat for the first time and you don't know anything about it. You're like, oh, I can just only eat wheat. And then you're going to wind up with 27 nutrient deficiencies as a result, and you're going to have protein nutrition at a minimum. And then you're going to learn your way out of that. You're going to be like, oh, I can eat some of my diet as wheat, but not all of it.
Right.
And so the incorporation of wheat into the diet wasn't that harmful. It was just this stupid way that they did it when they didn't know anything about it that went way overboard, that created an imbalanced diet. There are also reasons that led into the agricultural transition. Like, why did you invent agriculture? And it was usually a background of resource depletion that led to that change. Like, people developed agriculture out of necessity rather than choice. And because of that, there's unknown resource deprivation factors that went into that negative outcome.
Yeah. So I think we'd probably agree on this. Originally, us Homo sapiens, we were enjoying the fat of the land until we ate it all, hunted it all, foraged it all, and then we moved on. That's what we did. We kept moving and moving and moving. It was like, oh, awesome. We don't have to move.
Yeah.
We could till this land and grow stuff here, but we're just barely getting the hang of how that works. All right. If we get used to the thing that we're eating, I mean, it is sustaining us, right? But exactly what you said, things like stunted growth, I mean, we got plenty of data on that, on how that can be impacted by nutrient deficiencies. But nobody's thinking like that at that time. They're going, I need to be sure that I get my full complement of B vitamins, right? That's just not what's happening. So I could definitely believe that the hunting and foraging version of us was getting a better variety and a better complement of nutrients than the initial agriculture. So I think it makes total sense what you're describing. And then probably both through straight up boredom and trade, you end up with a greater complement of different nutrients that manage to come through in other ways. That to me makes sense as a broader story. But even then, why try that hard to find a single cause and then attach that cause? There's one thing, by the way, I wanted to be sure that I fit in here, because one of my favorite things that you said when I was like looking back on, I love when I happen to catch you on Twitter getting into a debate where cause comes up, because you sound exactly like me. And you already had a long tweet, but I just want to get to the money moment where you said you used the word, quote, linked, which intentionally introduces causal ambiguity and should be abolished from research papers with zero mercy. Just rejected entirely, and every instance of linked isn't deleted. So the reason I bring this up in this particular case is far too often, and in particularly in nutrition, people like to identify something ancestrally. They go, I see a link between this and this association here. I think that's what it is. That's what I'm going to call it. That's what I'm going to write in my book. That's what I'm going to have in my videos. This is going to be encapsulated in my grander theory of things, etc. And it's an interesting idea, something like what we just described. But it should be, even as we're talking about it, it should be relegated to, this is wild speculation, right? We should be comfortable saying things like that. By the same token, things like with nutrition, of those things that we can speculate, we should be just open about when they really are speculative and by how much they're speculative. I like that. I like more the Bayesian school of thought, and trying to be as thoughtful as you can be about probability of how right you are versus how not right you feel you can be.
Well, I think that there is a pathological fear about making causal inferences, and it's kind of polluted. You know, I even see people being afraid to make causal inferences about the outcomes of randomized controlled trials, which is not normative in current science, but you see the degree of fear like spilling over into that. And I think that it's a huge problem in the observational literature, because no one would really care about any observational literature if they weren't trying to read cause and effect into it. You know, there is some things that are best addressed with an observational study. For example, if you want to say how many stars are in the sky, you're not going to run an experiment. You're going to count the stars. That's an observational study, because you're trying to describe what is. But if you're drawing associations between alcohol intake and breast cancer, you're not trying to see if they're linked. You're trying to find out if drinking red wine is going to cause you to have more or less cancer. Like, no one would care about your study if you weren't trying to read cause into it. And so we have a big problem where people can't speak clearly because they know that there's something wrong with reading causality into their research, but they also know that they wouldn't be doing it if they weren't interested in the causation between the variables. And so you have this doublespeak that rises up where people use these really vague words like linked. But the problem with the word link goes beyond that, which is you didn't even say whether the association was positive or negative. And so I think it's incomprehensibly bad practice to use non-specific terms in science. Like, the purpose of an abstract, for example, should be to make the person not need to read the paper if they want your view of what the primary findings were. I'm not saying you should read abstracts and not read the paper, but I'm saying your abstract should be good enough that if I want to know what you think the main findings of the paper were, I should only have to read the abstract. So I shouldn't have to get the full text and go to figure 4B to find out whether the thing was increased or decreased. So if you tell me in the abstract that it changed, that's a bad abstract. It changed how? Did it go up or down? So I think the word link should be mercilessly purged out of scientific journals on both those bases, which is if you're studying something where the interpretation is critically opposite, if the thing went up or down, then you should say if it went up or down. And if you are trying, if you're trying to imply that there's causation there, you should be saying what it is. And if you're not doing that, you should also be clear about that. And so I just think that all this, we need to clean up our thinking about it. And the way we clean up our thinking about it is to clean up our language about it.
But and I'm not sure if this is exactly what I was reading from what you were saying. I feel like there's not enough fear of making causal inferences. I feel like causal inferences we're awash in causal inferences in nutrition studies, that they often are making statements that could be taken very easily in the abstract as confirmatory language, where I feel like they should be much more cautious with this was associated versus our study has a video where somebody was being critical because we said it was in the paper from last year. I forgot what word it was, but there was exploratory. We had exploratory in the results, but it wasn't in the abstract and it wasn't in one other place. Myself, because, I'm still very much working with a lot of people who are much more advanced than I am on this stuff and have done a lot more papers than I have. There's still a number of things that even spearheading the project, I'm learning about myself. I thought that was actually not a bad note, but by the same token, I looked at other studies this person had reviewed before in other videos and looked to those so that I could draw comparisons. How much did those other observational studies, particularly if they were exploratory, emphasize that they were exploratory in multiple places? And it turns out it was also either once in one of the paragraphs or not at all, where they would sometimes use confirmatory language. And one of those examples was the twin study, the Stanford twin study, the vegan one that also had like a whole series around it on Netflix or something like that, has very confirmatory language in it. It sounds very causal with the way that it states what it's saying, right? And I'm like, why is there such a concern with just saying this is hypothesis building? There's nothing. It's not a negative finding if you say, hey, this is something that we can build a hypothesis on, such that we can then go further steps where we can get more confident in doing something like an RCT. I feel like a lot of these observational studies almost feel this like need to have not only a strong positive finding, but that the positive finding can be taken as much more confirmatory and causal.
I think that we suffer from both. I think that's why there's such ambiguity and why you have really bad words like linked. It's that everyone's doing this research because they want to make causal inferences, and everyone feels that they're not allowed to make causal inferences. And so you have both of those things going on, and that does lead some people to make causal inferences too easily, but it also leads other people to use language that is sneaky and snaky by sort of wanting to imply a causal inference and not quite doing it.
Aren't the journals supposed to be the guardrails on that, though? in theory?
Um, I I don't think journals are very good at acting as guardrails. And I mean, you don't. I mean, so first of all, I mean, peer review is kind of a joke. I I did once have one good experience with peer review, where someone was very critical and changed my mind about some stuff. But the average experience of peer review is, as an author, I was taught to look for someone who looked like they had enough expertise in your area that they would pass as a reasonable suggestion in the eyes of the editor, but who would not know enough about the field to make a strong critique of your work. So that's the incentive for someone who's making their way up in academia who wants to get their papers published easily. As a reviewer, you're not getting paid for it, so your incentive really is to do the minimum. You can say that, you can put it in your paperwork that you reviewed so many papers, but no one's evaluating it. You're not being judged on it. You're not getting awards for it. You're not getting any credit for it. So your incentive is to do the minimum to pass by. But what I always thought was weird was that the average journal they don't have as a practice to have a statistician be a reviewer. So the average, like the stats in papers don't get a stats expert reviewing them. And there's no paper in any domain except causal inference that's getting a causal inference expert reviewing it. So I don't know how the journal is supposed to act as the guardrail there, because the editor is depending on the reviewers being the experts. The editor is the generalist. The peer reviewers are the content experts. So it's just not set up to act with those guardrails. And then, of course, there's plenty of debate about how to read causation using different methods as well. That's a whole field to itself. So if you did have causal inference experts doing the reviews, I think you would be opening up a can of worms.
I'm in favor, by the way, of all of that being transparent.
Yeah.
I think peer reviewers should sign their name to the reviews they're doing, and I think that their comments should also be a part of the publication of the paper. I feel like a lot of the problems with peer review is its lack of transparency. And I get what the argument would be. The argument would be that there would be less comfort on the part of, especially, say, less experienced peer reviewers to be honest and forthright. Like if they're reviewing someone else's work that, could possibly have retaliatory action against them in their career. But I think the trade-off is worse on balance.
The way it's done now, though, is the worst of all worlds. It's single blind.
Yeah, exactly.
So it really gives license to the reviewer to retaliate against the author, because the author has no idea who's reviewing the paper, usually. So it should either be double blind or single blind. I mean, one of the two. It should either be totally transparent or it should be double blind. But it is usually now single blind, which I think is the worst way to have it.
I agree. By the way, I do want to share a fun little anecdote, which is setting the stage. It's literally 10 years ago. It's 2016. For the very first time, I'm going to be doing a presentation on my very early beginnings of the lipid energy model. I've done some experiments. I've done some blood work by that point in time. But I'm really nervous because I'm going to be giving it to an academic crowd. And I was like, I wonder if I could just hire someone to review my presentation. And I found somebody I could have actually look at it. So you know who that person was? It's Chris Masterjohn. Do you remember?
Thank you for the honor.
Yeah. It was.
Yeah, I remember.
Yeah. So it was. What I liked was you not only reviewed it, but you also gave me some tips on presenting it. I had never presented an academic anything up to that point in time, and it was a fun experience. It was here in Las Vegas. You've probably heard of the Keto Gains group. That it was their conference. And it was at a station casino. There was, I don't know, maybe 40 people in the room or something like that. And it went really well. And then it was next year, this is the beginning of next year, that was low carb Breckenridge, which would ultimately become low carb Denver. And I'll remember this like it was yesterday. I was waffling between whether to try to present at low carb Breckenridge because it looked like I had a window there, or to try to take my deck to an NLA conference, which was going on at the exact same time, on the exact same weekend. The me of today would be like, duh, Dave, you absolutely need to go to Breckenridge. We're not going to get anywhere at the NLA conference. I'd, by the way, gone to the NLA conference since, like multiple times physically, but also virtually. And I know that the reception level was not going to be super high. But when I went to the Breckenridge conference, a lot of the same presentation that you had reviewed made it into that. And that ended up being the breakout moment for where the lipid energy model started actually getting some discussion. And it was in that same year that I ultimately codified the lean mass hyperresponder phenotype. And that was that's the beginning of pretty much all of it. At that point in time, if I'm not mistaken, you would have been considered not necessarily an LDL skeptic per se, but more, let's say, on the spectrum of skeptical as far as direct causal harm relative to even the average person in the paleo world. Would that be a fair characterization?
Yeah, I would say I believed that lipoproteins play a role in heart disease, but I did not believe that native LDL that has not been damaged played a direct causal role in heart disease.
And so would it be fair to say that right now, particularly given the materials I've reviewed, you do believe that there is a kinetic version of the lipid hypothesis, but it's with modified LDL particles? Would that be fair?
I've believed the same thing about this for probably the last 15, 18 years. And I've looked at new literature, but I don't think my model has changed much. But basically, I believe that oxidation of polyunsaturated fatty acids in the LDL particle membrane, doesn't have to be LDL particles, but as the kind of prototype, drives oxidation of the ApoB. Those things drive uptake in macrophages beneath the blood vessel wall. The reason for that is that the lipid peroxidation byproducts of the polyunsaturated fatty acids, seed oil fats, would otherwise kill the endothelial cells in the blood vessel, and the plaque is primarily a containment zone to prevent that from happening. And you could add some nuances to that based on newer literature, but they don't fundamentally change it. So the new thing is the LDL particle aggregation. And I actually had a back and forth on X with Thomas Dayspring about this, where he said, look, you're stuck in 1984, my friend. Everything that Daniel Steinberg discovered about oxidation of the LDL particles back then is not really relevant any longer, because we now know that LDL aggregation drives macrophage uptake regardless of oxidation, and so you don't need to have oxidation, and so therefore you're wrong. There doesn't need to be any damage. They'll aggregate. But if you look at the research on aggregation, first of all, one of the primary drivers of aggregation is oxidation of the LDL particles. So really, we're just back where we started. And I do think that there's a nuanced layer you could add to it, which is that the saturated fat to choline ratio in the diet is probably a negative for LDL aggregation, because phosphatidylcholine is the primary anti-aggregation moiety of the LDL particle, and that's completely dependent on choline, which is an essential nutrient. If you don't eat it, it's not there. But that doesn't really add anything to my overall dietary philosophy, because I've long known that the dietary saturated fat to choline ratio is a major driver of fatty liver disease. So it's like I've added one more reason to believe that the more saturated fat you have in your diet, the more choline you need. I've, juxtaposed what I believe about fatty liver onto this LDL aggregation. But the LDL aggregation doesn't negate at all the importance of oxidation. It enhances that thread. And so, my model's updated a little bit, but I don't fundamentally believe that what's happening is something, at its core different from what I believed 15 years ago. And there was a period of time, very briefly, before you and I had ever talked, where I was an LDL skeptic, where, the LDL skeptics don't believe in the role of lipoproteins in heart disease at all, basically. Before you and I had ever talked, I had settled on a middle ground where I thought, yes, there's a lot of convincing evidence that lipoproteins are involved in heart disease, but their involvement is not one of the concentration in the blood driving the heart disease process. It's one of the degree of damage happening to them is driving the plaque. And that, of course, is moderated further by the fact that plaque progression is not the main driver of whether you have a heart attack or a stroke, but it's actually plaque degradation that is causing most heart attacks and strokes. And so I think there's a lot of confusion about that, because it's very hard to measure the actual primary determinants of plaque degradation, and coronary calcium isn't really one of them, although it might generally correlate with the degree of the process going on.
Yeah, that's a lot of that seems to be one place where everyone seems to agree, which is that the calcium is more going to be an association of the disease development itself, but is itself not part of the degradation of the plaque.
Well, I think that is wrong, though. It's the reason that people believe that is because you can't measure the calcium that matters. So the calcium that comes up on the coronary calcium scan are the macrocalcifications,
where it's more stabilized,
where it's either irrelevant or maybe it contributes to the stability of the plaque. The microcalcifications in the fibrous cap make it more, the mechanistic evidence shows that, which you're not going to get in humans. The mechanistic evidence shows that microcalcifications that don't show up on imaging, and you can know this because you can, for example, in an animal, not a human, you can just, take the tissue and analyze it for calcium contents. There's multiple different ways that you can measure calcium, some of which are much better than imaging. And so my mechanistic model does involve a role of calcification of the fibrous cap, causing it to be more brittle and more likely to degrade and cause a heart attack or a stroke. But I don't disagree with the people who are saying what you're saying, because I think they're coming from a clinical background and they're saying, well, none of the calcification that we're measuring is going to be the determinant of whether someone is going to have that plaque break and drive a thrombotic event. And so, yes, I agree with that. I just wouldn't throw the baby out with the bathwater on the calcium.
I'll talk you through my model.
Okay.
I want you to feel free to pause or interrupt me at any point.
Okay.
I don't know if you've heard me talk it through in this podcast before, but it's pretty close to where it was, I want to say, starting at around 2018, 2019 in particular. But one thing that I loved was, even in 2019, I have lots of tweets, I've got lots of receipts, I was really driving home the relevance of transcytosis. It has never made sense to me. I'd be curious to know if you disagree with me on this one. It has never made sense to me that there was a passive process by which lipoproteins were forcing their way into the subintimal space where there wasn't active inflammation, because the issue is that those endothelial cells, they've got nice tight junctions that are 5 to 10 nanometers wide at most, and they're like a zipper between all of those different transport proteins in between. But on top of that, it's also the nature of the way by which the blood flow is going. They're not coming like straight at the endothelial wall to hit those junction gaps quite as easily. The mass of the lipoproteins, it makes no sense as to why they would just happen to passively seep through them. You would expect other elements to also come through them as well. But this all changes at a site of inflammation, where of course they're going to separate from each other intentionally. That's part of the playbook, like, with regard to being able to allow for monocytes to come in via VCAM to then differentiate macrophages. Anyway, so the junction gaps not in a site of inflammation in healthy endothelial cells, I don't see lipoproteins going through there, but I definitely see lipoproteins going through in the very thing I was talking about at the beginning of the podcast, which is through transcytosis. SRB1 that I was just mentioning, that's one of a few different ways, but probably the most prominent one that's studied right now for how an endothelial cell through an active process is literally grabbing a non-modified LDL particle, non-modified, and chaperoning it through to the other side. There's the idea that LDL particles could actually smash through an endothelial cell to the other side was always ridiculous. That always never made sense. They would all be Swiss cheese.
I'll make a quick comment and then let you keep going. The research that I've seen indicates that it's not just inflammation, although what you said about inflammation illustrates why the tight junction permeability is a dynamically regulated process. So you should expect it to be dynamically regulated by other things. But one of those things is the amount of parallel force generated by blood flow. And at the branch points, you get less parallel blood flow because you do have a more irregular pattern of blood flow. And those are the points where you have more atherosclerosis, and those are also the points where animal experiments show that you can have gold nanoparticles up to 100 nanometers going through. So I disagree on that, but I agree with you on the contextual regulation of it.
And here's what I would speculate, but I can't say I know for sure because I haven't looked into how granular that they've gotten with endothelial cells at those branch points. I would expect, just as in any other place where there's a greater degree of shear stress, you would find a higher activation of inflammatory markers in particular because of the fact that they are under a greater degree of mechanical stress. So, in other words, would I, for example, expect that there's greater apoptosis of those cells? I would expect that they probably get damaged more often because of the mechanical stress of where they're not only situated, but coronary arteries, as you know, if you're picking places of intensive hemodynamic stress, you can't, that's one of the tops that you're going to find. So endothelial cells that are there, they're in a class 5 hurricane all the time, right? And branch points are one of the toughest spots of that. So I would expect that the immune response is pretty busy there. It gets calls to have to go to that part of the neighborhood very often. And
yeah, I think we're probably largely in agreement, and I'm not trying to take inflammation out of the model, but there are regulators like nitric oxide that I think are kind of iffy how you want to classify them. Like certainly nitric oxide is involved in the immune response, but it's not a classical immune. You can have plenty of nitric oxide signaling that is unrelated to the immune response. So
it's definitely part of the vasodilation cycle. There's a name for that, but it's where it's doing the squeeze down, where it's actually the expression of it for coming in and out to be able to facilitate the blood moving down. So I would expect, definitely nitric oxide expression is very relevant towards atherosclerosis.
Yeah, well, I think we're largely in agreement on that.
But yeah, it's not that I am expressing that I'm sure I'm right with regard to the hemodynamic stress of those branch points. It's that I want to be convinced that I'm wrong,
right,
with regard to that aspect of it. But as far as there being just a sheer greater degree of junction gaps at places for which atherosclerosis increases, technically I'm in agreement. I just think that the reason for the higher amount of the gaps probably is related to that threshold of risk with regard to where those areas are. And it makes sense in that regard with the pathology that we see with atherosclerosis, right? You would expect that if it was strictly about permeability in a concentration gradient, that, hey, greater and greater concentrations, well, that's all throughout the vasculature, right? And if you want to take it more from what I think is a more defensible version of the lipid hypothesis, which is it's more of a multiplier. Concentration gradient is the gun that's getting loaded, and the other risk factors are what's pulling the trigger. They're multipliers. Okay, that's an interesting discussion, but then there's other directions we can go from there. But let me bring it back to where your and my models may overlap, which is with regard to lipoproteins that get modified, right? So I definitely believe the immune response attends to modified lipoproteins. Like, that's literally why we make, LOX1 and CD36 and other receptors whose job it is to bind to modified lipoproteins. Where this gets interesting for me is I believe that ApoB-containing lipoproteins are actually part of the innate immune response. I think you even have written in ways that are in agreement with me on this.
They are.
For one, getting to one of our mutual favorite subjects, alpha tocopherol, otherwise known as vitamin E. It's often described as though it's the oxidative defense of the lipoprotein, where I think you would agree with me on this. I believe it's host defense. I believe that the fact that we have it sitting there protruding the monolayer of the lipoproteins as part of the antioxidant capability for coming into contact with other reactive oxygen species in the lumen suggests to me that it's host defense. I mean, either you're getting it there or you're getting it potentially in a cell wall where you can have an oxidative chain, right? That, to me, I think, is a value for having ApoB-containing lipoproteins in the lumen, particularly whatever concentration gradient it is. But also, it's relevant in the extracellular space. It's relevant in the intima, right? So if you have transcytosis, like I've just now described, if you have endothelial cells who part of their playbook, when inflamed, are actually expressing more LDL receptors, are actually doing more transcytosis through SRB1 in order to bring them into the subintimal space, it makes sense to me that that'd be part of the innate immune response if one of the benefits of that are the things lipoproteins can bring, such as alpha tocopherol, into that space. And especially if macrophages have an easier time taking those up, particularly if they do things like, bind to pathogens than otherwise. So in the way that you just now described it with regard to a foam cell, and I would describe this as part of that dog pile of the innate immune response, potentially, that, yeah, I think that there are probably a lot of what we might think are going to be pathogenic plaques, foam cells that are building up, that the immune response is often taking care of just fine. We just never see those. The ones that we end up seeing are the ones that really have gotten out of control, right? And sure enough, they tend to be in the places like the branch points and so forth, where the immune response is having a tougher time and where we can make the environment externally worse through what we're doing with diet, what we're doing with sleep, what we're doing with exercise if we don't value recovery time enough. That, to me, is more of the model of atherosclerosis. I think that we have a long list of ways that we can create damage to endothelial cells. I think a lot of it's just the cost of doing business. I think you've got natural apoptosis, for example. I think you've got, and that's just kind of standard. But if you've got a good diet, if you're not nutrient deficient, if you've got good enough recovery time, all of that, right? Then you probably don't have a vasculature that's at high risk, relatively speaking. and this is my hypothesis, I'll emphasize, um, regardless of what the concentration gradient of the lipoproteins are. It may even prove to be beneficial to be at a certain level of lipoproteins. I don't know exactly what that would be. I don't know, what the curvature is of diminishing returns or anything along those lines, but that's the key question.
I think that the primary purpose of the lipoproteins is to transport stuff in the blood. And so vitamin E, for example, mostly is transported through its entry into VLDL particles in the liver to the rest of the body. That's like the primary way that you would get vitamin E anywhere else. And it's primarily taken up, at least in endothelial cells, through selective uptake from HDL particles. And so I don't think you need any other explanation for why it would be found in an LDL particle. And so I'm a bit I'm not saying it's not playing a role in the immune response, but I'm a bit skeptical that we need to explain its presence in lipoproteins that way. That's just how it's getting distributed.
I actually just have to interject.
Yeah.
It's you were the first person through one of your, I don't know if it's a podcast. I think it was a podcast. This was literally, I want to say 8-9 years ago. You were the one who was describing I think it was the helix protruding from the monolayer. And then it was from that I ended up looking this up. So, and granted, I realized this was a while ago, so it may not be fresh in mind because you were like studying up on it at the time. And I don't think it was to make the point that it was doing exactly what I'm describing for the host defense. It might have just been for its own defense with regard to being an antioxidant for the defense of the ApoB lipoprotein. But I'm not even kidding when I say you're the one I actually learned that from.
I do think that there are always going to be multiple roles of something, and redundancy is a positive, desirable part of any kind of engineering project. So I'm not saying that vitamin E does nothing in the lipoprotein other than get transported. I'm just saying that at the most fundamental reason for why vitamin E would be found in the lipoproteins is because that's how it's transported throughout the body. So it needs to go in the lipoproteins no matter what. And
right.
The vitamin E that gets taken up into the brain is coming primarily from HDL particles, delivering it to the endothelial cells of the blood-brain barrier, taking it up. And I've written about this a long time ago. In fact, the first thing I ever published that was indexed for PubMed was a critique of the I don't remember if it was torcetrapib or it was anacetrapib, but it was the CETP inhibitors were killing people, and they were attributing it to off-target effects. And I said, you guys should look at the vitamin E transport, because if you block CETP, you're blocking the transport of vitamin E between the lipoproteins, and you're preventing the HDL-dependent delivery to the endothelial cells. But I definitely agree that LDL particles are part of the acute phase response, and this is supported by the fact that before the historic decline in infectious disease, familial hypercholesterolemia genetics were associated with lower mortality rates, and that can be explained by having higher concentrations of LDL in the blood. And that does add a quantitative inference to what you were saying, where you said, I don't know where the threshold is. The threshold is that it's most effective in familial hypercholesterolemia range. So I do think that the quantitative range on that is at the point where just, it looks like it's a trade-off being at that point. If you're vulnerable to heart disease, it's in the heart disease risk range. But if your primary vulnerability is you're going to die of infectious disease, that is where you want it to survive that. And then one other thing I would say is, I don't agree that I don't think that the dose gradient of the particle concentration is very relevant.
Okay, but
Even as an amplifier, because if you go back to the cell studies that Steinberg had done in the 80s, in the late 70s and 80s, what they basically showed was that macrophage LDL uptake, which native LDL uptake in a macrophage occurs through the LDL receptor, it goes like at super low concentrations, there's a concentration gradient, and then it just flattens off. And if you look at the concentrations where the concentration influences uptake, it's like orders of magnitude below what occurs in human plasma. And then you have LDL modification, which is primarily lipid peroxidation. You have a 5-fold increase in what gets taken up into the macrophages. So at the concentrations that are relevant, macrophages don't take up LDL particles dose dependently. But they do take up 5-fold more
right
when it oxidizes. So I think there's a lot of confusion generated by the fact that randomized controlled trials of statins can't tell you anything that teases out the causality of the native LDL from the oxidized LDL. So the lipidologists think that you can make causal inferences about the native LDL concentration from that, but they're using I believe what they're using that as a proxy for, in that population, the native LDL concentration or the total does act as a proxy for the amount of oxidation going on. And I think the statins are reducing the amount of LDL oxidation that goes on by clearing it out of the blood faster. And so I think that's led to a lot of confusion about the dose gradient.
So a few things to unpack there. One of my favorite studies is on in vitro macrophages from those who have homozygous FH versus the normal controls. And it had both those who had homozygous FH untreated and those who were treated with LLT, lipid-lowering therapy. So it's really 3, right? The macrophages that were untreated had a different expression of receptors. I can't remember. I want to say one of them was CD169 or something along those lines. But it seemed as though it had tried to compensatorily, it tried to offset what it couldn't accomplish with its LDL receptor with these other receptor activities, right? But what I liked about this study was it was kind of fitting into my model as to why it is more relevant that those who have homozygous FH in particular, one of the big 4, one of the, the monogenic FH levels, right? So it's LDL receptor is what Brown and Goldstein were originally looking at. There's ApoB, there's PCSK9 gain of function, and then there's LDL RAP1, I want to say. Those big 4 both result in super high levels of LDL and also result in very early atherosclerosis. And so they became, a real catalyst point where people were going, oh, okay, well, it's given. I mean, this is Bradford Hill's strength criteria through the roof. But then,
hold on a second. So Bradford Hill, no one should ever talk about Bradford Hill without reading his 1965 paper.
Totally agree.
Bradford Hill never claimed that the strength of an association was evidence of causality. He said that and by the way, what he said was strong was 200-fold increase. He said his example of a weak correlation is a 2-fold increase. Yep. And what he said was that when it's that big, meaning a 200-fold increase instead of a measly weak 2-fold increase, if there's a confounder, you're probably going to know what it is. Whereas if it's a 2x increase, then the risk is that there could be many confounders that are hiding right under your nose and you can't see them, right? So his point wasn't that a 200-fold increase in something means it's causality. his point was that a 200-fold increase in something means that if it's not a causal association, you're probably going to see with red flags the other 200-fold differences that could account for it. And so in this case, going back to how Bradford Hill might have analyzed it, where he's sitting at this table with us, and we were to educate him on what we know about lipoprotein metabolism, is he would say, yeah, there's something there, but is it the native LDL or the oxidized LDL? There's no capacity to tease that apart.
Well, and this is where I would bring it up. You're probably familiar now with that there's studies on comparing monogenetic FH with polygenetic FH and no known genetic component. So they're looking at populations where they match up the LDL levels.
Okay.
And those with monogenetic FH, the hazard ratio, so the no known is reference at 1. The hazard ratio for the monogenic FH is 2 something. I want to say 2.1 or 2.0, something like that.
For lifetime?
I don't remember what the time.
Okay, well, anyway.
But assume lifetime, just to be generous,
Yeah.
right? The polygenetic was 1.3, right? So again, matching up the LDL levels. So is there an independent association with atherosclerosis for those who have monogenic FH separate from the LDL concentration levels? We know the answer to that now. Brown and Goldstein did not, which is the answer is yes. There's something distinct and independent about having monogenic FH apart from the LDL levels. Now, I was on a podcast with Simon Hill and William Cromwell. And I brought this up. I brought up also the in vitro study with regard to the those with monogenic FH who have homozygous FH, and why I think that's highly relevant. To steelman their position to pick up William Cromwell's position, he was saying, well, they're, children with homozygous FH, they're also developing in the womb with this difference in the LDL. And so that might turn out to be relevant as to why they might have more advanced atherosclerosis than somebody who might have just picked it up the same levels at, like, say, age 50 and then lived for another 3 years. Why they wouldn't see the same level of atherosclerosis as, say, the children of Brown and Goldstein.
I'm not quite following what you're saying is distinct about the monogenetic. So what is the distinct
monogenetic? So let me let's Brown and Goldstein, they were looking at the LDL receptor in particular.
Right.
Homozygous FH, it's both alleles, and therefore there's just no binding capability.
Right.
Therefore, there's a higher concentration of the LDL particles in the bloodstream. And that is thought, by looking at the higher levels of LDL in the bloodstream, that is what is causing this early atherosclerosis in those children.
Right.
Right.
So what's distinct about the...
When looking at the outcomes of risk, as far as like earlier cardiovascular disease.
Right.
That...
Well, heterozygous FH in general is you're not getting it when you're 18 months old, but you have a higher risk when you're in the 40s and 50s.
Right.
And 60s, and then it starts closing.
Right.
And so what are you saying is distinct about but you're now saying that the heterozygous you can break into people who have... I'm sorry, no, I don't follow what you were saying about what's distinct about the mono. You were saying the homozygous FH with the same the two alleles is distinct how?
Is distinct different in that it's independently higher risk than what the LDL levels would predict with the lipid hypothesis.
Than someone without the genes.
Correct.
Oh, yeah, but then I agree with whoever was steelmanning the other side that can easily be explained by the lifetime area under the curve.
Right. Let me pose the same thing back to you, and let me see what your answer is. Pretend I want to replicate Brown and Goldstein's children.
Okay.
I know that the little
ones with homozygous
FH.
Okay.
So let's say that I know a guy who can change his LDL cholesterol to homozygous FH levels.
Okay.
So that the cumulative LDL that he will have over, let's say it's not even 3 years, let's say it's 5 years, right, will be comparable to the little girl that Brown and Goldstein were looking at.
Okay.
same exact cumulative levels of LDL, would you expect that he would therefore have a comparable level of atherosclerosis development?
Not when he's a kid.
When he's an adult, let's say
okay. So first of all, to be clear, I disagree with Brown and Goldstein that the causal variable here is the concentration of native LDL particles in the blood. So I'm not trying to support that. I don't agree with it. But I do think that the impairment in the LDL receptor is what's driving the atherosclerosis. And I think that the way that it is driving that is by increasing the residence time of the LDL particles to unreasonably long amounts, which then leads to their modification, which then leads to all the other stuff that we talked about post that.
And this might be a difference with our model. So my model is, I think it matters literally to the immune response, to the macrophages. I think that's very relevant. I don't know how much it's specifically the macrophages, but that's a nucleated cell that does take up unmodified LDL particles as well as modified LDL particles as part of its immune response. And I think that it's very relevant.
Well, it's a macrophage. What is it doing besides an immune response?
I'm saying its immune response is modified, right? With regard to the relevance of the uptake of unmodified LDL particles.
Yeah, I mean, it's a macrophage in the subendothelial space is by definition engaging in an immune response by being there, right? Okay,
yeah.
And so yes, so everything that it's doing is part of its immune response.
Right.
Yeah.
So going back to, do inflamed endothelial cells transcytose greater numbers of ApoB-containing lipoproteins?
Okay.
We know the answer to that is yes.
Okay.
The EAS 2020, and that's why the whole field of lipidology shifted in 2020 as EAS consensus mechanistic paper said, yeah, transcytosis now.
Okay.
And that's why I thought it became super relevant. And that's where I bring it back to the immune response. I believe that higher level of transcytosis is part of the playbook of the existing immune response. I believe macrophages taking up not just unmodified LDL particles, but also, not just taking up modified LDL particles, but unmodified LDL particles is part of the immune response.
Well, when a macrophage does it, yeah, but pretty much any cell is going to take up some LDL particles.
Sure.
Yeah.
But how is it that
But by situating itself in the subendothelial space and becoming resident there, it is engaging in an immune response at that location by definition. So everything that it does there is, I mean, any of these cells are going to take up some LDL particles from the blood for the most part. So
here's the distinction to make between us. Do you, if we could stop inflamed endothelial cells from transcytosing non-modified LDL particles, do you think that would be a good thing or a bad thing?
I don't know if I've seen all the relevant literature, but my gut instinct is that it wouldn't matter that much.
I think it does matter.
Okay, what?
I think it's because it's part of the dogpile effect that's typical of the innate immune response. The innate immune response is get all the guys there, right? And
well, where is it going in your model?
They
transcytose it to where?
It's transcytosing to
and
the
why?
subintimal space.
Okay.
Because
why?
There are multiple things ApoB-containing lipoproteins can potentially do in that space, such as bind to pathogens, such as, I believe, the alpha-tocopherol and the trinols. All of those are, I think, participants in the process of also turning reactive oxygen species into non-reactive products. And importantly, I think that they have a capability to bind to things that are harder for the macrophages to pick up, making it easier for the macrophages to pick up the lipoproteins that get modified.
Well, if I were to incorporate that into my model, I would say that would be a good thing.
Correct. I think it's a good thing.
Well, when I said I don't think it would make a difference, I meant I don't think it's going to make more pathological plaque development.
But could it possibly be a net benefit for support for those macrophages to accomplish their goals?
I think developing the plaque is a net benefit, because the alternative is that you got a bunch of toxic crap that's floating around the blood.
And in the subintimal space.
The best. Well, yeah, but it's got to be in the blood to get into the subendothelial space. The best outcome is, well, the best outcome is that you don't have these things that you need to protect against, like lipid peroxidation and infections. But given that you have them, the best outcome is for if an LDL particle is oxidized, gets taken up by the liver. But if you have, that assumes that it's circulating. And so if it's stuck in the subendothelial space and it's oxidizing, I think the next best thing after that is that the immune cells take it up. And I think they're involved in taking up anything that doesn't belong anywhere. And I think the alternative to the macrophages taking it up, if the liver doesn't take it up and the macrophages don't take it up, then it's going to create more toxicity. And so I think the plaque development is the best thing in that situation. And it's, the simple development of some plaque, I don't think is really a problem anyway.
Wait, yeah. So it sounds like we're a lot, because I kind of want to divide these two categories.
Sure.
Category one is bad stuff gets into the subintimal space, right? So something hemorrhagic, where something's now in the subintimal space that's neither a lipoprotein or a macrophage yet, but that gets the cascade of the chemokines, the inflamed areas, like, come on, immune response, come here, right? The two things I just mentioned, the junction gaps are opening. Hey, that's one way in which both monocytes turn into macrophages, but also how lipoproteins could get in. And then there's the controlled process by which they're being brought in. So we know two ways in which lipoproteins get to this place where there's pathogens, right? But then there's this other one that you talked about. The second category is the lipoprotein, possibly in the lumen, very often in the lumen, gets peroxidized. It gets modified before even talking about subintimal space, right?
It could, although there's a lot more peroxidation that happens in the subendothelial space because it's a more oxidative environment.
Right. In either of those cases, now we're not even talking about, it may not even be at the level of an immune response, because we already know macrophages are down there anyway. There's a lot of them that are, you could argue, are doing janitorial duties. It's not at that level for which it's a site of inflammation.
Everything by default is dynamic. So you've got monocytes that are going in and out. And there's no reason that an LDL particle oxidized in the subendothelial space can't leave.
Correct.
I mean, people act like their reading of the mechanism is so poor because they act like the diagrams they're looking at are static pictures,
and
but they're snapshots of dynamic processes, right? So there's no protein that has a binding affinity of infinity for anything, right? And so if something gets bound, some of it's going to get released, and if things get in, they're going to get out. So by default, you assume everything that goes in comes out, but it can stay there. And if it does stay there and bad things are happening, then bad things that stay there are worse for that place than bad things that happen and leave.
Yeah, so it sounds like our models are going to collapse, because what I love is what you just said is exactly what I've said on this podcast so many times, and puts us at odds, by the way, with a huge number of people out there who believe that when a lipoprotein gets bound to a proteoglycan and then gets modified, it's just a one-way ticket. All that's going to happen is macrophages are going to get there. They're going to just be stuck. They're going to start. I mean,
these people aren't serious. But unfortunately, and to be honest, I think the reason mechanistic speculation gets such a bad rap is because people are so bad at it. And I like a lot of people, I'm like, yeah, you shouldn't be mechanistically speculating.
It's funny. I love hearing this, because I often say this and I don't mean it to sound as bad as I realize it probably sounds, but I constantly go, as an engineer, and I'm constantly thinking in terms of dynamism, because why wouldn't you think that? Why would you? Okay, we've learned macrophages are pretty good at moving. They're really good. In the subintimal space, they're crushing it, right? Why do we think they stop? Why would they engulf an oxidized lipoprotein?
It's because people forget chemistry after they leave it, and so they're leaving behind the things they just like basic principles of you've got a population of molecules and they have an average kinetic energy, but there's a distribution of the energy, and some of them are higher in energy. They're all moving around, and the probability that they interact with any other molecule to have a reaction is a stochastic process. And if you forget that, which people do, I mean, because the way that science is once you pass that test and you get into the next class, you forget a lot of it. But then once you're in your career, you focus in such a niche area that it's very easy to forget a lot of foundational principles. Otherwise, there's no. And then I do also think like the tools that we have at our disposal are, I mean, I think we're coming to a place where we might be able to teach with animations very well, but a lot of the existing animations are just have really low fidelity to the actual processes involved. People are mostly looking at static diagrams in textbooks or in papers, and they're just forgetting the dynamism that came with the original foundational principles from chemistry. So
I think there's another component though that's difficult to work around, which is that the process of atherosclerosis is on long time horizons, and it's in a place where it's very difficult to study. It's hard to study the development of plaque in the arteries very easily. You can't just get a little video camera in there and be like, okay, now let's watch it. And 12 hours later in your animal model, you've got this perfect pathology slide by slide of exactly how it worked and why. You've got to go through the process of sacrificing. You've got to try working things out through, sonography and all kinds of other ways in which you can try to tease things out. But even then, you've just got these issues where in order to get plaque at a level that you can study it more effectively, even in animal models, you're usually inducing it to such a degree of complexity that I'm already being skeptical as to whether the immune response can attend to it in the same way that I feel very confident is being attended to all the time, every day. I feel like we're developing tiny, in the same way that we have scratches on the outside of our body, and that's managed and taken care of quite easily. I believe it's the same thing with the vasculature. The catch with the vasculature, and where the engineer in me gets excited, is it's actually a very difficult engineering problem. It's a scarce space, and it has to be absolutely open at all times. Like, imagine a freeway that you can never close, right? How would you work out all problems that can come along the freeway, the sides of the freeway, exactly how best to manage and work around it?
You're reminding me now that the early animal experiments on atherosclerosis that were done up to the point of 1932, when Nikolai Anichkov he did a massive English language review of a bunch of studies that were done over the previous 40 years that were mostly published in other languages. And one of the big findings there was anything that induces atherosclerosis, if you just take it away, the atherosclerosis goes away. And so the big finding from the early animal experiments is that atherosclerosis is temporary and dynamic, and that if you removed the cause of it, you wouldn't have to do anything to get rid of the plaque. It would go away itself. And that seems obvious to me, because the purpose of the plaque is really a situational thing,
right?
Where you're like, this is the best we can do in this moment in time. It's not the best long term, but it's, over the course of this short time horizon, it's better than the alternative. And the reason that plaque progresses in humans, as you showed in your study, the primary determinant of plaque progression is plaque, which is another way of saying once you have some plaque, you're going to wind up with more. But that's because you have not removed the conditions that favored the plaque in the first place. And so I think if you favored those conditions and then you disfavored them, you would see a reversal.
So I'm glad you brought it up. We should probably chat about it anyway. It's a hot topic these days. At the time we're recording this, because it'll probably get released afterward, is we're a little less than 3 weeks from the film that was filmed around the study. How much have you kept up on the KETO-CTA study?
I read the tirade that Nick put on X and the quote that he tweeted from you. I looked at the papers, but I didn't, it seemed to me like there was much to update. So
Yes.
I looked at the preprint, but I was like, well, I'm not going to spend too much time on the older papers if
Probably
they need to
smart.
totally revise the
Yeah.
yeah. So I don't know. I'm interested in what comes out of it.
Yeah. So I do want to sort of set the stage in let's start with, at the time that we're recording this, most of what needed to come out has come out. You actually may be the first person who I'm recording after it's been, I was literally able to make it official that we requested the withdrawal of the paper that contained the Clearly analysis, and the journal accepted, and notably nobody objected to this. The journal didn't object, and Clearly, who provided us the analysis, did not object, which says a lot, right? But this data set, my lawyers are allowing me to say, is anomalous. Nobody seems to be coming out in disagreement with it, but it created a very interesting situation, a kind of curveball. So with that said, let's pretend for a moment that it's stricken from the record. We're going with both the scans that nobody seems to disagree were properly done, and the analyses which nobody seems to be in disagreement were also properly done outside of Clearly, and who all agree with each other. That includes the semi-quantitative, that includes the Q-angio, and that includes heart flow, which also is an AI-guided reading. So they all support the original thesis of the paper, which is two things. One, plaque predicts future plaque progression. So if you had baseline plaque, it was more likely that you would get more plaque. And the other is that there's no association between LDL and ApoB and plaque progression. So those lipid levels, when I say that there's no association, you would know what I mean when I say this. I mean, we're talking R.01 and R.015, I think, something like that, right? Depending on which analysis it was. R squared, I think. Anyway, the plaque predicting future plaque is not a strong correlative. So if you do have plaque, it's not a given that you're going to have a lot more plaque progression. We had people who had plaque that regressed. In order for us to have people who had plaque that regressed, they had to have had baseline plaque, right? But there is something that I can share with you here, which is a paper that's going to, I might get in trouble, but I'll put it this way. I'll say that we did more advanced statistics. Are you familiar with what's known as minimal detectable change, MDC?
I couldn't do it myself, but
okay. So this is what it is. Given a distribution, you can figure out what amount of a change longitudinally from point A in time to point B in time there needs to have been, what amount of a delta, a distance there needs to have been to feel a 95% or greater confidence that there was a change at all, right?
Right.
In our cohort, that's 5 people. So when we ran more advanced statistics, there's a level of noise between progressors and regressors that we can concede up front. We don't know how many are true progressors versus regressors below the noise floor, right? That might sound frustrating, but it's actually kind of awesome. It means that we just have such low levels of plaque in the folks that we have that even with the longitudinal data, it doesn't provide us a lot of information on the change, save 5 people, which would be expected in an otherwise low-risk group. But it's also what necessitates why we want to get these 5-year scans. We want to see how many we can re-recruit to get a third set of scans. Third set of scans being 1 year from their first means we'd need to start in October of this year. And that data would be gold.
And they need to stay on the diet.
I don't care if they stayed on the diet. I just want to be sure that they report to us if they did. I mean, don't get me wrong. I would love if as many as possible stayed on the diet, but I just want the data.
Right.
Some number.
Well, I mean, I guess it'll be useful to the extent you have more people who stayed on the diet.
Exactly.
Yeah.
So let's say, for the sake of argument, let's say that we re-recruit. Let's be conservative. I hope it's conservative and say 65. And let's say of the 65, 30 stayed on the diet. So I'm being super conservative. Sounds like a really small sample size, particularly given we started with 100. But is it that small of a sample size if we still have an average LDL of 240, 250 with a spread that's in the hundreds, right? No, that's actually really good. That's really good for testing the simplistic version of the lipid hypothesis. The simplistic version of lipid hypothesis, we should see a signal. We should see an association between LDL or ApoB and plaque progression.
Well, it's testing whether the assumptions of the lipid hypothesis apply to the population that you're looking at.
That's why I like to say the simplistic version of lipid.
Right.
So the simplistic version of the lipid hypothesis would be, it doesn't matter what population you are. High LDL, high risk in general. That's not to say it should apply to every anecdote. I'm not being that guy. Yeah. But at a population level, even in a population of 30, would we expect to see a signal?
Right.
We would. We would definitely expect to see an association, given what the present modern version of the lipid hypothesis is, where applied to every population.
Yeah.
So,
well, I'm excited to see that data when it comes.
Yes. I tell you, the challenge. I mean, I'm writing a book on this because I want to tell the story, the broader story. The movie touches on it, but as you may have picked up, it's engineer from the outside goes, gosh, guys, let's just do a study, and gets together a mixed group of people, mixed hypothesis group, and with, I would argue, the best of intentions, thought if I could just go raise the money and then hand it off to the research center. All right, you guys are the experts. You got this from here. And that won't be how the book goes. The book, I go through the account of a lot that happened. Once you're inside the sausage making, it's a different story. In getting your PhD, I mean, you've had a chance to do some experiments yourself. And also, I'm correct on that, right?
Yeah, I have.
And do you have anything that's associated with the new business that you've got? I assume you have.
Not yet.
Not yet, but you're working on that?
Yeah.
Okay,
yeah.
Cool.
We are. Hey, can we do a bathroom break?
Oh, of course. Yeah. Yeah, you're having coffee. I'm surprised you didn't want it sooner. Okay. Let's get to one of the greatest hits.
All right.
Seed oils. Something fascinating is that I tried through all 3 of the COSI conferences. We've held 3 total for fundraising for our research. All 3 times I tried to get a live seed oil debate going. I failed all 3 of those times because I couldn't get somebody who was a proponent of seed oils to come to the conference. To be fair to them, it's a charity fundraising conference, so I couldn't pay for their travel. But I had no lack of people who wanted to argue the other side of it, who wanted to argue that seed oils are not great. I've also offered, there was a breakdown in a debate that was set to happen between Paul Saladino and Lane Norton to take place. I know
about that one.
Yeah. That one didn't happen. But there were some conditions that, given what I know now from behind the scenes, I can understand why Paul Saladino bowed out. But I offered up. I said, hey, I'll pay for you guys to come here and debate it here at
And they
the
said no.
Yeah. Well, Paul Saladino said yes. Lane Norton took a pass.
Right.
And I think I could put it out right now. I think I could. I mean, the catch is that I do still want people who, have at least some prominence, some background, some knowledge, at least, not just somebody out of nowhere. But I am impressed by how much the concept of being anti-seed oils is kind of broadly dismissed. But there doesn't seem to be an interest by people in genuinely engaging in like something like this, a real sit down, deeper conversation where we really break out the data, where we really discuss it at length, right? And I'm not even necessarily a hard end on either one of those two sides. But you're one of those voices that's been talking about it for a while, right? Like, when did you first start discussing your concerns with seed oils?
I don't remember, maybe 2004, 2005.
Wow.
I mean, certainly 2008 or 9-ish, I had written quite a bit about it. But yeah, it's been a while.
Now, I'm going to do my best to steel man the other side,
Okay.
but I'll concede up front that, again, I don't really have. I'll say what my own opinion is. My own opinion is I think it's going to be a dose makes the poison thing. I think if I have some soy or canola oil in my salad dressing, it's not that big of a deal. I do agree that probably if it's the base fat for my keto chow shake, that's going to be a problem. That's my guess.
I take it you eat more keto, you drink more keto shakes than you eat salads.
Correct. Yeah, that'd be my gist. Do you disagree? Should I be concerned about the canola oil in my salad dressing when it comes up?
I mean, I guess it depends how much salad you eat, but you just said you don't eat much salad. So, I mean, the dose makes the poison is kind of obvious for any poison.
Well,
but
let's have fun.
yeah,
Let's say I have a Caesar salad every day.
All right.
Right? One Caesar salad every day. Would you speculate that would probably be?
I think if you eat something every day, it should be made with the highest quality ingredients you can get. And I don't think seed oils are them.
Gotcha. Okay. So what are the problems with seed oils?
Well, the main problem with seed oils is that they increase the amount of vulnerable fatty acids in your membranes that become a risk for tissue damage. And that's something that will naturally get worse over time as you age. And that's because your mitochondrial function is declining as you age, which means less fuel available for the antioxidant defense system and all of the other systems that are going to protect cell membranes. And as a result of that, I think that aging is a sufficient risk that you don't need to say, well, seed oils could be a problem if you smoke, or well, seed oils could be a problem if you have mercury toxicity, or well, seed oils could be a problem if you've had an industrial accident and you're exposed to too much herbicide. You know, all those things might create an acute problem that makes things really bad in a short-term time horizon because you've been eating canola oil as your main fat for the last 15 years. But that's not the base case. The base case is aging is going to be your primary problem when it comes to increased reactive oxygen species, decreased mitochondrial fueling of antioxidant defense, etc. In which case, the being loaded with seed oil fats becomes a problem. That's my position.
So yeah, let's unpack that a little bit, and you correct me where I'm wrong here. So we have throughout our body, we're lipivores, to borrow Amber O'Hearn's version. We have fatty acids, and fatty acids are they're long, and if they're saturated, then there's no double bonds, right?
right?
Those are like butter. Butter's high in saturated fat. It means at room temperature, it can remain relatively solid. However, you then have mono and polyunsaturated fats. Mono would be a single double bond. Poly would be two or more double bonds. And they're floppier. They've got a hinge where that double bond is. And the problem with that double bond is it's more susceptible to being oxidized, right?
Close. So the problem is the carbon that's situated between two adjacent double bonds. That carbon is the carbon that is highly vulnerable to peroxidation, which is why it is only polyunsaturated fatty acids and not monounsaturated fatty acids that have significant vulnerability to lipid peroxidation.
So when we're eating them, we're bringing them into our body. And it's worth noting, you're not anti-polyunsaturated fatty acids. We have essential polyunsaturated fatty acids we have to get in, right?
Well, I called them precious yet perilous in an article that I wrote a long time ago. And what I meant by that was they're needed in very small amounts, but they become a liability at higher amounts. And that's pretty common. Like there's lots of things that you need in your body, but you only need small amounts of them.
Like minerals. Or
sure,
specific, like, okay,
Even
iron might be a
vitamins,
good
but-
example, right? Yeah. So so I saw a video that I liked where you talked about seed oils, and you drew a distinction that I've drawn before with other things that I really love, which is that you called it that polyunsaturated fatty acids are not the cause of oxidative stress, but they are an oxidative liability.
Yeah, that's right.
And I like the way, go ahead and unpack that for us real quick, because I like the way that's phrased.
Yeah. If your house was totally made of glass, it would be more vulnerable if someone threw stones at it. But if no one threw any stones at it, it wouldn't be that much of a problem. So a liability is something that is an increase in risk that you're accountable for. But it's not straight up damaging, right? Like you are building your body out of things that are more easily damaged by things in the environment. And so that doesn't automatically make you have a disease you didn't have before, but it makes you more vulnerable to those stresses that can damage those fatty acids, and it makes your tissues more vulnerable to that damage.
And so this is also why you bring up the issue with a lot of these studies that seem to be supportive of seed oils tend to be very short in the time that they've been studied, whereas the ones that say otherwise had very long spans of time. I think you said like 5 to 8 years from like the 50s, 60s, and 70s. Is that right?
Right. I mean, this is another big problem in the randomized controlled trial literature in general, which is there's not a lot of things that have been studied over 4 to 12 week time horizons, as well as 5 to 8 year time horizons. But whenever something in nutrition is studied in both, the trend is that things look good in the short term and they look bad in the long term. And you could also see that with drugs. So for example, SSRIs are mostly studied for 8 to 12 weeks. There's none that have been studied for 5 years. But the average person who's put on SSRIs is on them for 5 years in clinical practice. And if you look at the observational literature, you know, the trials say that they're pretty easy to get off of if you want to get off of them. But the observational literature says that it's a lot harder at 1 year than it was at 12 weeks, and it's tremendously harder at 2 years than 1 year, and it's catastrophically harder at 5 years than 2 years. You know, so this is a general problem across the literature with all kinds of things that we use, which is that we make long-term inferences based on short-term trials. But it just so happens that the trend is really clear with the seed oils, which is that there are things they do over the short term of 12 weeks that are good. You know, they're positive for liver fat. They're positive for metabolic control over the course of 8 to 12 weeks. And over the course of 5 to 8 years, it looks like they are either neutral or bad for heart disease, and that they increase the risk of cancer. And so if you're looking at that from a longevity perspective, heart disease and cancer are your top 2 killers that you're trying to avoid. But, so the gist of it is that if you have something that's primarily going to cause harm by sitting there and waiting to be harmed, then you're going to need to have older populations and longer trial periods to see negative effects, as opposed to something that is toxic, where you might be able to administer it for, an hour and see an acute negative harm from it. And so that's a problem, because so many studies are done in young people for short time periods.
Yeah. How do we solve that? How do we incentivize for longer, because it costs money, right? That's going to be the pushback is, hey, this is going to cost so much more money than if we do something that's in a shorter term.
Well, this is a way bigger problem in science, which is that people do studies because they're practical instead of because they are of interest. And so, I do think that the vast majority of research is a waste of time. And
it's a strong statement.
And so, I think it's true, because scientists, in the mythology of science, scientists tell themselves that what they're doing is they're looking at the existing research, they're identifying the gaps in our knowledge, they're ranking those by importance, and then they're studying the important gaps in our knowledge to get us to the next advancement. But what they're actually doing is writing grants to stakeholders that they think will get funded based on how easy it is to get those based on the resources that they have at their disposal. And the various stakeholders involved in the funding, it's rarely is that ever organized in a way that the stakeholders who fund science have a systematic view of what the knowledge gaps are. They usually have their own priorities. And so it often, in practice, what I see in academia is people are dancing around the edge of the current health recommendations. And because they also have an incentive to not disrupt the funding, their reputation with the funding mechanisms, right? So they fancy themselves as being pioneers and explorers of knowledge gaps, finding the ones to conquer and conquering them. But in reality, they have a timidity and fear of the grant overlords, because they know that they don't want to rock the boat of the public health establishment too much. And so what they do is they wind up doing, they had a company that was willing to give them their wild blueberries. And so they do a study on wild blueberries and how they increase total antioxidant activity in plasma. And then they say that we now have a novel reason to support the existing recommendations to eat 5 to 9 servings of fruits and vegetables per day. And so there's like, you could make a meme out of it, of like what the scientist thinks he does, what the grant funder thinks he does, what's really happening, and I think that's the state of it. So a lot of people are just, I want to study seed oils. I want to learn something about fatty liver disease. Let's do another 12 week trial of seed oil fats,
Right.
And it's just, they know they're going to get funded. They know that it's like a novel reason to support the existing recommendations. They're not rocking the boat, but you're trying to play this delicate balance between, I'm doing something novel and I'm not doing anything novel. I don't want
Right.
my
wanna
novel to be a threat.
Right. You don't.
But I can't pride myself as a scientist unless I do something novel, right?
Right. You're trying to prove something new is happening, but not something too disruptive, which is it's just crazy. And here's the thing. What you described is actually something I'm very interested in with regard to a diet very high in polyunsaturated fats. Because how much of it is compositionally different from what is a typical ancestral diet that we've had before, such that it really does change the composition of the bilayers of our cells, is both fascinating and terrifying at the same time. Because if you were to take all of the fatty acids of the phospholipids that are in our cells and then throw them into a pie chart, right? Large preponderance of them would be saturated. I myself don't know these numbers off the top of my head as far as how many are monounsaturated, but a decent proportion would be monounsaturated, and then a small fraction would be poly. And to be sure, we need all three, right? There's areas of the cell, and depending on it, you're trying to hit this sweet spot of rigidity and fluidity with all these cells. But your diet can influence that pie chart. It can expand that slice of the poly on those phospholipids. And the same thing I was talking about with like the floppiness, that you can look at the how that actually changes the composition such that the cell then itself is different, right? So getting back to the analogy that you had with if you build your house of glass, right? So some of my house is glass on the outside, right? You could throw a rock and there's a chance it could hit one of the windows. But yes, if I just kept, shingle by shingle building it back into glass, that would be very meaningful, but it would take time. So if I'm eating a diet that's very high in one particular fat that would get so far as to structurally change my cells, that's not going to happen overnight. That's going to take some time. But it would be very interesting to find out how much that becomes relevant toward the human health long term. I think it was a podcast with Kate Shanahan, and she may even have it in her book. I think her book's Dark Calories, but also gets into seed oil concerns, that there's, I guess, research that gets into this, how much a lot of modern Western populations have a higher proportion of polyunsaturated fats in their cell bilayers. Are you familiar with this literature?
I haven't looked at it for a while, but yeah, there is data on that.
So,
I mean, in general, we have more omega-6 and less omega-3. Probably more total PUFA, but I haven't looked at it for a while. And of course, it's going to depend where you look. Like, you're going to see a much bigger difference in adipose than in different tissues and different compartments in tissues are regulated to a greater or lesser degree. So in the brain, for example, you're going to have very strict regulation of certain fatty acids because they're very important. So you have very strict regulation of brain levels of DHA and arachidonic acid. Arachidonic acid is a complex omega-6 fat that is most abundant in the diet in liver and egg yolks, but which you can also make from the linoleic acid that you find in small amounts in traditional fats or in seed oils. And DHA is mostly gotten from fish or from egg yolks if the chickens eat insects. But you can make it from omega-3 fatty acids from plants as well. But the diet is more capable of changing the other fatty acids in the brain, not so much those, but its diet is a lot less able to change fatty acids in the brain than elsewhere. And it's very hard to change fatty acids in the brain in an adult versus an infant. But out in the rest of the body, you're getting, very big changes in adipose tissue if you're feeding seed oils over 4 years, and you're getting modest changes in other membranes. And so some of those have been measured and some of them haven't, but you'll get a lot more data on red blood cell phospholipids than you would get on, liver cell membranes or something like that,
Yeah, and I think I would expect this with red blood cells given their half-life, right? I would likewise expect greater cell bilayer change with adipose tissue given its elasticity. It's more likely.
You're also getting like
triglyceride content changes though. So like the adipose triglyceride storage is not well regulated because it's just excess energy. And so you definitely, if your cell's trying to regulate the fluidity of the membrane, then it's going to change its composition somewhat in response to diet because it's meeting that goal with whatever it has available. But there's an intrinsic resistance to changing it too much. And so those fats are diverted into other places. And the biggest change is going to be in the adipose triglyceride content. But that does not mean that there's no increase in the seed oil fats elsewhere. It's just they're more modest. And one of the harms that happens over time is that the vitamin E gets diverted into the adipose tissue. So you might have a modest increase in the tissues that are, tissues of this, let's say the cell membrane fatty acids in the skin. You might get a modest increase there, but you also have a decrement of vitamin E available to the skin because you've had the biggest change in adipose tissue, and that's diverted the vitamin E in that direction. But yeah, these changes generally are going to depend on the tissue, and they're also going to take like 4 or 5 years to play out.
That's going to be a long experiment.
Yeah. Well, I mean, to take this back to what's practical, this is why first of all, there are some questions we're never going to have settled because the trial that was the longest out of any to date has been the LA Veterans Administration Hospital study, which was concluded at the end of the 60s. And the main conclusion from that study was that their trial wasn't long enough because it was 8 years. Most of the seed oil trials done at the time were 5 years. And so they said that, and the reason they thought that was because the time-dependent changes in their study showed that things were looking worse and worse as time went on. But a lot of those negative changes, like the increase in cancer in the seed oil group, didn't even start happening until you started to see some changes around 2 years, but it was really in the 5 to 7 year mark that the incidence of cancer really started increasing. And there was no change in total mortality the whole time, but there were some inklings that at the end, the total mortality would have started worsening in the seed oil group. And so they looked at that and they said, well, if we really wanted to see the true effect, we really have to run these trials well in excess of 8 years rather than 5 years, as has been the typical goal. And so the conclusion circa 1970 was we need trials well in excess of 8 years. And what did we get? We got a lot of 7 to 12 week trials.
Right.
So if it hasn't happened now, I don't really see it happening in the future. And so there will be gray areas that you have to navigate with the existing trial literature, and you have to do your best with what you have available, and you're going to have to incorporate your bias somehow. So if your bias is ancestral diets, you're going to you're going to come to a very different conclusion than if your bias is in favor of technological innovation. Which of these do you take as your primary inspiration to how you interpret the literature is going to put you in very different places. And a lot of people aren't comfortable with that type of ambiguity, but that it is what it is. So I can tell you what I think the literature says, but my position is colored by the fact that I am skeptical of something that never existed in the human diet before. And that skepticism does color how I view the literature. But what the literature says in broad strokes is there's a bunch of observational studies that I completely discard because they have super small effect sizes and they were all started collecting data after there was a massive global campaign to say that everyone should be eating seed oils if they want to be healthy. So to me, that's so biased that I don't even look at it.
Right. We're back to healthy user
And
bias.
then we've got these short trials and we've got these long trials, and I prioritize longer, larger trials, and I prioritize double-blind trials over trials that were not blinded. And there were only 2 long, large, double-blind, randomized controlled trials of seed oils
Ever. Right.
ever. Well, I mean, in the years-long time frame, right? So there's double-blind, randomized, 5-week
Right.
or whatever,
Right.
right? But out of the ones that were 5 to 8 years, there's only 2 that were double-blind and randomized. That was the LA Veterans Administration Hospital study and the Minnesota Coronary Survey. And the gist of those 2 trials, when you put them together, is seed oils probably bad for heart disease, probably bad for cancer, probably not good for anything. And, you know, I could give you a much larger view of every other trial that was done, but I come from the ancestral diet position, but I also know my way around evidence-based medicine. And I know that double-blind, large, randomized controlled trials are better than trials that weren't blinded, trials that weren't randomized, or non-interventional studies. And if you know your way around Bradford Hill thought that a 2x association gave you no causal information, and the seed oils have a 1.15x in an obviously biased context, you know. So that's how I see it.
I've had a hypothesis for a long time that we'll see, and it could. And these AI has been fascinating with regard to research, because you can pose a number of research questions. You have to be fully cognizant of the fact that it can hallucinate all kinds of things, so you still have to follow up, catch into its references and so forth. But it has allowed for smarter searching than it did before. And so there are a number of questions like the one I'm about to bring up now that I'm like, oh yeah, this would be one to go ahead and hit the, I like to hit multiple LLMs at the same time, by the way. But I think that some threshold, I don't know how much, some threshold of the lowering of LDL from things like seed oils may play directly into what you're describing with regard to greater peroxidation susceptibility of lipoproteins, because they're going to be cleared faster with scavenger receptors. Like they're not going to have the same half-life. How much of those are actually directly picked up, for example, by things like LOX1 on endothelial cells, I couldn't say. How much of them, there's another component too, which is some amount of PUFAs, and depending on the type and depending on the circumstance in the liver, will be more preferentially heading in the direction of the ketogenesis pathway relative to the relative to esterification or assembly with regard to ApoB containing.
Well, that's why the
lipoproteins.
liver fat goes down.
Yeah,
Because
right.
they're oxidized faster. But what you're getting at is also the fundamental mechanistic trade-off of the seed oils with respect to heart disease, which is this is why you want to do trials on them, because they do regulate in the liver, they are more likely to be esterified to cholesterol, and so they decrease the free cholesterol, but they're more likely to be oxidized than to go into triglycerides. So they decrease the liver fat and they increase the ketones, because oxidizing fatty acids is what generates ketones. But in the LDL particle, they make it more vulnerable to lipid peroxidation, which is the main lipid peroxidation of the polyunsaturated fats from the seed oils in the LDL particle is, even according to the conventional lipid hypothesis, when the scientists involved bother going into the mechanistic detail of it, Daniel Steinberg, he passed away a few years ago, but he was the chair of the 1984 consensus conference where NIH concluded it's proven, we know that cholesterol causes heart disease. And he named his book The Cholesterol Skeptics, the Cholesterol Wars. And the subtitle was The Skeptics versus the Preponderance of the Evidence.
Right.
And his chapter 4, I believe it was, he said, in order to really get the smoking gun, in order to clinch the argument. We needed to explain the mechanism of how a high concentration of cholesterol in the blood causes the plaque to develop.
Right.
And he says that the mechanism is that the polyunsaturated fats in the LDL particle membrane oxidize, which oxidizes the ApoB, which stops it from getting taken up by the LDL receptor and increases its uptake by scavenger receptors on macrophages, which then creates the plaque. And then he goes on and says, now that we have shown the mechanistic detail of how a high concentration of cholesterol in the blood causes heart disease, now we can move on to how to treat it, which is the statins. And
I didn't know this.
And it's even funnier because Steinberg became a corn oil skeptic because of this. And so he was one of the people who very quietly replaced seed oils with olive oil. And this was kind of like, with the LA Veterans Administration trials showing the increase in cancer, and people like Steinberg, who were like chief architects of the cholesterol hypothesis, being afraid of the lipid peroxidation in the LDL particle, they kind of very quietly moved away from promoting corn oil, which is what my grandparents were told to eat, and moved towards olive oil because it was considered neutral, because it doesn't affect cholesterol levels, but it doesn't cause peroxidation, right? But what's funny is that Brown and Goldstein wrote the foreword to Steinberg's book.
Oh, wow.
And they said that everyone should be eating seed oils. But anyway, so the point is that everyone believes this mechanistic story when they're really pressed, except Thomas Dayspring, who's like, Steinberg is, he was back in 1984. Now we know it's aggregation, but like I was saying before, it's the same thing. So the fundamental trade-off that you're evaluating here is, yes, you're going to increase LDL receptor activity and you're going to clear the LDL particles from the blood. But while you're doing that, the LDL particles are going to be enriched with the fatty acids that form the mechanistic basis for how they get damaged and wind up in the plaque. And so with that trade-off, that's why you can have conflicting results even in trials. And that's why you got to be careful in how you evaluate the trials, because you're dealing with a double-edged sword. But that is ultimately why you want to do the trials, because if you're saying like, my primary weapon against this disease is something that has two functions, one is to prevent it and one is to cause it, then it matters a lot which one plays out in the long run.
Right. Yeah. I've been a longtime critic of studies that and this comes up with pharma all the time. They set up something for which it's a fixed amount of time that's announced, but then they ultimately make a call based on events. So if they reach a certain amount of a threshold of statistical significance, they go, we got to stop here. We got to stop here because now the placebo group should be on the intervention because we have a statistically significant difference. It'd be unethical for us to go any further, right? And that new phenomenon, I believe, is unfortunately resulting in a lot of extrapolation. So in the same way that I like to bring up, you can take a couple of stocks, Apple versus Microsoft, at given different points in time and be like, one is statistically significant than the other. That one's going to win. In fact, if we just extrapolate out, we see that Microsoft absolutely crushes Apple forever and ever and ever, past this certain time.
Well, it's funny because if you were actually making a portfolio, you would assume mean reversion as your base case.
Yeah.
I mean, I'm not saying you can't take a point of view that one outperforms long term. You could, but your baseline assumption in finance is you would like if you're trading the Bitcoin to gold ratio, you're gonna rotate it in and out of it based on a mean reversion principle. Unless you think you have inside knowledge that one of them is going to outperform for a particular reason that the market doesn't understand. But but that actually does work in health. Like in general, when you see large differences appear in a short time horizon, you actually do observe mean reversion as the predominant long-term outcome. Like if
like if like the
I think
the
some people
probability
may not be following you on mean reversion.
Okay,
Like, unpack that for a sec.
Mean reversion was developed way back in the day when it was observed that although fathers who are tall obviously have the genetics for tallness, their sons tend to be shorter than them. And that's because how tall you are is a combination of your intrinsic likelihood to be tall based on your genetics, as well as whatever other random influences that were available at that time to push you up to a higher height. And so what you see is that there's no inexorable physiological, real biological basis for why a tall father would have a short child. But the principle of random chance leads to that being the average outcome, because some of the tall fathers were taller than their own genetic potential, and that's why they were tall. Some of the fathers with tall genes were shorter than their potential, but they are not they are not counted as tall fathers in that cohort, right? So but the big realization was that if you looked at the data backwards and like if you looked at the sons and you pretended they were the fathers and you looked at it going the other direction, it looked exactly the same. And that showed that it wasn't any kind of biological transmission of anything. It's just that when you see something high, you expect it to be lower later, because those are the principles of random chance. So and this is why you have randomized control groups, because if you take a bunch of people with high cholesterol, you will see it go down over in the second measurement, no matter what happens, if your sample is large enough, because some of those people always have high cholesterol and they're true high cholesterolers. But there's a variation in your cholesterol throughout the year. Like it's going to go up or down 20 points. If you measure your cholesterol over and over again in the year, it's going to go up and down by 20 points on average. So 17 milligrams per deciliter is what you'd expect.
I have some opinions on that, but
so
go ahead.
Well, okay. But if you take, let's say you take 500 people who you define them by their high cholesterol. If you just follow them over time, the average of the second measurements should go down. That doesn't mean that some of those second measurements aren't going to go up, but if you keep repeating that over and over again, the average of the second measurement goes down, and it's because.
So let me see if I can restate this to help you out. Let's say I wanted to specifically get people with high cholesterol. So I go down the road to a room. I've got my PTS diagnostics, and I'm going through the room, and there's 100 people in it, and I want to grab 50. And they go, guys, I need to grab the 50 people in this room that have the highest cholesterol. I'm going to get all of your numbers, but I'm going to take the highest 50, right? And so then I do that. I get the highest 50. And sure enough, they're only just barely a little higher than the average of the other half, right? And I'm like, cool, got it. What you would argue is that a year later, what it turned out was all I really did was I grabbed a bunch of people who at that time were on the higher end of average, but probably closer to the other half of the room than just the speckles of few people who have persistently high LDL. Right. And therefore, it's more likely than not that the mean average might be lower.
Well, that would be more likely to be true the higher their cholesterol was. But
compared to the expected average of the group of people in question. So if I went to a room that was full of people with heterozygous FH, it'd be different, because they literally have that genetic predisposition for the higher LDL.
Well, yeah, but no, it would be exactly the same, because if you define people by their high cholesterol, some of those people are that's their average cholesterol, and some of those people they're higher than that average. And it's also true that's like you have a random spread. So if you took, like, let's say you took everyone and you didn't define them based on high or low. Some of them are going to go up, some of them are going to come down, and some of them are going to stay the same, right? You're going to have variation all over the place. But if you select for the people that have high cholesterol, you are enriching your sample in people
that have high cholesterol because they always have high cholesterol, as well as people who met your criteria because their cholesterol was higher than it usually is.
I don't know that we can know that, though. We can't really know that.
You can know that because you selected them based on how high their cholesterol was.
Right.
It's definitional. It's
right,
definitional. If you select for, let's say you look, every performance of everything is a combination of its intrinsic ability to perform and luck, right? So if you select for high perform, like let's say you just like select for successful people, you're getting a combination of people who met your threshold for success because they were talented, but they met that threshold through a combination of their talent and luck, right? So your threshold is getting enriched in luck by the fact that you only selected high success, right? Because everyone's success is a combination of talent and luck. Every person's cholesterol in a given moment is a combination of their average cholesterol and how high or low it is compared to that average. Like this is it's true across.
But with one snapshot, we don't know the true average. Take it to the successful.
No, you don't
people.
know the true average. That's why you accidentally enriched your sample in people that are going to go down over time.
But take it back to the success people. We don't know, for example, how many people actually would have been more successful. We happen to catch them in a bad year, right?
Yeah, that's true.
So getting back to the cholesterol.
That's true, but the people who are disproportionately off their success mark are disproportionately less likely to wind up in your enriched selection of the top.
Yeah, that's, I guess, given the
anyway, we can leave this, we can table this discussion.
Yeah, we'll table it.
But the point that I wanted to make from it is that whenever you see a huge effect in a first study, it is almost always supplanted by less impressive studies later on. And it is almost always the case that if this later studies are larger, they're even less impressive. And so when you see dramatic things, they usually aren't as dramatic as you first thought they were because of the principle that everything tends to regress to the mean. So the point is, when you have a short-term big effect, if you have any sense in your mind, you believe that it would get less impressive over time, right? Now, you happened upon a great finding when that's not true. When something gets more impressive the more you study it, fantastic, phenomenal, right? But to add the slightest dose of cynicism to the reading of this ethics sham, they know this. They know that if the results look really good at a year, they don't want to risk them looking not as good as that. And they know that the principle of reversion to the mean makes it more likely that they got lucky than that they actually stumbled on something that everyone must be given now. So therefore, they have to cut the study short while it looks like everyone must receive this now. And then they can make everyone receive it now, which is in their profit motive.
I think it's, me personally, I think it's the statistical interplay. I think that you can, okay, so good example is we know that there are lipid lowering trials like, say, Fourier, that had more people who died in the intervention arm versus the placebo arm, right? But it was not statistically significant. Was there a risk that if it went longer, it would have been statistically significant? Sure. Can I say that I know that that's what would happen? No, I can't say that. But would there be a reason if you had some statistical significance on the cardiovascular side, particularly if it's within a subgroup, I forget if it was the above 100 or the below 100, to then say, let's stop it right here before we see the all-cause mortality shift
It's also known that subgroup analysis is usually wrong.
Yeah, well, that's where it also gets a lot more interesting, right? Is how often there's a study that, I mean, this is part of the problem. Again, getting back to just how this whole thing works is you have these studies that I had no idea, just the sheer magnitude of money that goes into these things, these trials. But at the same time, there's this other factor that concerns me, which is that everybody who's a part of these trials knows that they're never going to get replicated, right? It's never going to happen. You know, if you're part of a giant trial that's going to study a certain thing, particularly if it's a certain drug or a certain device, and there's not just tens of millions, but possibly hundreds of millions that are going into it, there's zero chance there'll ever be some scientist over your shoulder being like, I want to double check that. I'm going to replicate the 4-year trial or something like that. It's not going to happen ever, right? That part concerns me a lot. It makes me care a lot more about ensuring the objectivity of all components involved with the management of the trial. But, you know, this is where I genuinely want to, like, I'm literally working with scientists who are in the field, who run these kinds of studies, and I want to give it all the benefit of the doubt. But Chris, if you've been following, I have my reasons to not give benefit of the doubt to providers, especially of measurements, to say, yeah, this is right, trust us.
Right.
Particularly if after the fact I can't get them to even do a quality control check. So bottom line is, it's a challenge for me because now, having been on the inside of the system, I can't say my trust has gone up. It's maybe gone a bit the other direction.
Yeah. Well, I'll be interested to read all the drama if you ever are able to disclose all of what you found about the Clearly analysis. But I do think that like we were talking about before, I do think that we need a lot of creativity in reimagining the incentive structure. But there's a lot of political inertia, especially around NIH funding. And, you know, one thing that I learned from talking to people who are active in the MAHA part of the administration is, for example, NIH doesn't even have much control over NIH funding, because there are so many fiefdoms set up by bureaucrats around their corner of the NIH funding that they would actually need Congress to reapportion funds.
Really?
Yeah.
Huh.
And so I talk to people who are in NIH, who are kind of observing that process, and their outlook was basically like, need a lot more people in Congress on our side to really make huge change in that. So I think that there's a big problem with the political inertia that has the control over most of the funding. And I think it might be hopeless to try to use politics around that. And I think we should, but I could see much faster results through trying to establish decentralized crowd-funded science. And I think that's we definitely do not have a strong link between people who systematically review the existing literature for the important knowledge gaps without contamination from pharmaceutical interests. And I think that, in principle, medical professional organizations would be well suited to do that type of work, but they are so contaminated by the pharmaceutical interests themselves that they can't. And then there's not much organization, and the talent for systematic reviewing is not really there, or it's not, I'm not saying it's not there, but it has not been built up into the infrastructure that's needed to create that. And maybe you're doing that now, but you know, we need too many of the people who are the most talented at doing systematic reviews of the literature are taken up by the big organizations that are kind of, you know, very embedded in all these malincentives. And so we really need to recruit that talent, and it's multifaceted, right? So that means people that are that talented in a statistics point of view, people that are that talented in a causal inference point of view, people who are that talented in the content and the history of the field.
Well,
I think that's the direction that we need.
I mean, okay, there's a couple things. One, obviously, I'm a fan of crowdfunding research. It's hard. It's really hard, but I didn't have any strings attached to me. I could just do what needed to get done for the most part. The major curveball that I ran into was, again, just I had to invest trust, in the institutions that we were paying to do the thing that we needed to get done. The accountability issue is humongous, but one important thing that I am very proud of is that early into this research, I established the creed that we were just going to keep being open. We release our code, we release our data. We've done it with every paper that we've done up until this point. We release the data, including the semi-quantitative and clearly data, a portion of it, not all of it, but of what our principal investigator would allow us to put out. Since we're doing a third set of scans, there's reason to not put it all out yet until we get all the way to the end of what may be the extension of the study. But it's my intent to put literally everything out. And that's actually hard to do because many of the researchers I'm working with are working for me on a song. I'm saying, hey, come do this research with us. It's for the betterment of science. And they're like, that's great. As long as we get to publish, and that's great. We'll get a whole bunch of data. We could maybe, make 12 papers out of it. But if I go, well, actually, I'd like to release all the data just as soon as we publish the first one, it's disruptive to all of the potential publications that they could put out. But this gets us back to what we were just discussing. Why is that so important to researchers to publish papers? Well, it's the old publish or perish model, right? How important are you as a researcher? Well, that's in large part by how many papers you put out, what your H index is, how much you cited versus not cited, and so forth. There's not a huge incentive for running studies and then releasing all the data.
Well, the incentive has to be built into the journal requirements.
Right. Agreed. And so I've heard that it's moving that way.
I've seen that it's moving that way. I've seen journals that require that.
I've heard that the NIH is moving in the direction of trying to require,
they should,
as part of their grants, that all data are released.
Yeah, they should.
They absolutely should, because it's taxpayer driven, right? We should have access to the data that we paid for, right?
Well, CDC shouldn't have any private data either.
Agreed.
Yeah.
So I feel like a lot of the concerns that I have would evaporate in this day and age, especially of AI, where data could be open and released and the code could be out there and we can run our own analyses, and especially it makes it easier to find problems. It's not going to be any secret. In the book, I had a local LLM and I was on top of the Clearly data, and that's why I was able to find problems within 24 hours after I received delivery as president of the Citizen Science Foundation. I was supposed to get the raw anonymized data. I got it. I saw the issues. Thank you, AI, because I'm not, an advanced biostatistician, but I was able to find these issues pretty quickly. And again, nobody out there is defending it. And I think it's because they also have access to AI. They can take the data that we made open and available to them, throw it into the same thing, see the same issues. And it's why we're at where we're at now. Transparency, in addition to that level of access for capability, made the accountability real. And I think we'd have the same thing with all of these other studies. If there's a study that seems very odd, unusual, it's harder to get away with changing things, fixing things, altering how one thing works or another without the fingerprints showing up in the way that the data is reported. And that's why I think we have to have more transparency.
Agreed.
You look like you're fading a little bit. Am I right?
It's up to you.
No.
We can,
We've gone a 2 hours 58 minutes.
I'm good to keep going.
Good to keep going. Here, let me do this. Let me get to like a few of the questions that got posted, because I asked if people wanted to ask you anything. Actually, it's funny, we just mentioned this one. So this is from Mark Biskey. It says the FDA's definition of food is food. Does nutrition science have an actual definition of food? And if so, what is it? If not, doesn't that make nutrition science the only branch of science that either does not or cannot provide a definition of what it is the science? Show more. It doesn't show the whole thing. But that was one of the questions for you.
I would say the definition of food is something that's edible.
I like
also nutrition science isn't food science.
Yeah. I like Gary Taub's term, which I've now used a lot more, which is that there's food and then there's food-like substances.
Yeah, I mean, a lot of things that are. I'll be honest, I don't see the value in defining food. I suppose if food is the equivalent of what should I eat, then I think you should define that. But I just don't agree that food is synonymous with things that I should eat. Like, there are foods that I shouldn't eat.
Yeah. Speaking of which, literally the next question: what does Chris Masterjohn eat? And supplements. What supplements do you take?
My diet right now is based on a blend of beef and organs. So I've got some liver, kidney, and heart in there, and I've got some straight up ground beef. I've got a little bit of rice. I've got a little bit of nixtamalized corn tortillas. I've got 4 or 5 egg yolks a day. I've got some potatoes, some tomatoes, peppers, and broccoli, some olive oil, and quite a bit of cheese. And average diet's about 150 grams of carbs a day, net carbs, about 160 grams of protein a day, and the rest fat. Oh, and I got about 150 grams of heavy cream a day. And supplements, I take 8 grams of creatine. I take 600 micrograms of molybdenum. And I take 1 capsule that has 600 micrograms of K2, 100 milligrams of CoQ10 as ubiquinol, 1 capsule of food-based vitamin C, and about I think 3.3 grams of cod liver oil are my supplements.
I'm interested in the vitamin C. Why do you, and what's that mean? Food-based vitamin C.
And I've got 100 grams of strawberries a day, which are also good sources of vitamin C, and are contributors to my carbs.
Wait, in addition to everything you said,
Yeah.
You eat 100 grams of strawberries a day.
Yeah, that's right.
Wow. Okay.
Yeah.
So explain.
Is this face for the precision or for the strawberries?
What's this for the? Well, just
for the amount of food.
The distinction. Yeah, 100 grams of strawberries is like it seems very distinctive. It's not.
Oh no! I mean, I eat 50 grams of white rice and 104 grams of corn tortillas, and a quarter pound of U.S. Wellness organ blend with a quarter pound of beef. Like, I got it all grammed out.
Yeah.
Yeah.
Last one. So
about 2,815 calories a day. right now.
How many?
2,800, 15 calories.
2,800. So I'm probably around 2,400.
Yeah,
But I think I'm less active than you are.
maybe.
Can you comment on ancestry DNA versus 100% genetic sequencing from DNA complete for COMT findings?
I would only use whole genome sequencing.
All right.
I wouldn't bother with the SNP chips.
You followed the whole 23andMe thing. I was
Not closely, but
yeah. I downloaded my data before they dissipated. But where can people find you?
they can find my newsletter at chrismasterjohnphd.substack.com, and they can find my mitochondrial testing at mito.me.
Well, thanks for coming, Chris. This has
Thanks for
been
having
a
me.
blast.
Yeah,
All right. Maybe we'll have you back again sometime.
cool. All right. Thanks, Dave.
Cheers.